this will be the programme for the day. Just to say a little bit about what the Dundee Root Medal is and where it came from. So the Root Medal is an award, a legacy award funded following the International Society of Root Research Conference that held in Dundee in 2012, and it's hosted by the James Hutton Institute, and this year is the 11th award of the Dundee Root Medal. So the award recognises outstanding contributions to root research, celebrating scientists whose work has significantly advanced our understanding of root biology and its impact on global agricultural systems, sustainability and food security. I will say at this point that this is my first year as chair of the Dundee Root Medal Committee, and I would like to take the opportunity to thank Professor Tim George, who was the previous chair. Tim has been instrumental in keeping the award going and taking us to the point we're at now. Year on year, it's continued to grow, and I'd like to acknowledge the role that Tim has played in doing this. Fear not, he will not be leaving the Dundee Root Medal Committee, just stepping back a little bit, but he'll still be an integral part of the committee. So thanks again, Tim, for everything you've done and continue to do. So as I mentioned, the Dundee Root Medal is a legacy from the ISRR conference held in 2012. Last year's winner was Amelia Henry. The year before that was John Hammond, and the year before that was Malcolm Bennett. The inaugural lecture for the Dundee Root Medal took place in 2015, where the medal was awarded to Professor Jonathan Link, and it highlights the value of this award and the recognition of people who I'm sure all of you appreciate have led significant areas of research in this area. So on to 2025. So I'm delighted that this year the Root Medal has been awarded to Professor Simona Radetoiu from Aarhus. So Simona holds a PhD in plant physiology from the University of Agronomical Sciences in Bucharest, and has worked on the topic for the last 2 decades, building collaborative and internationally recognized research at Aarhus. So Simona's a professor of plant molecular biology and genetics, with her research exploring the molecular mechanisms that govern plant microbe interactions, with particular focus on the signaling, recognition and accommodation of beneficial microbes inside roots and in their rhizosphere. So our work has led to landmark discoveries of key plant receptors involved in root nodule symbiosis and revealed the molecular strategies plants use to distinguish between beneficial and harmful microbes. Now, these findings have significantly advanced our understanding of how plants engage with symbiotic microbes and how they contribute to the assembly of complex microbial communities in the rhizosphere. So Simona's research has been funded and supported through national and international funding bodies, and she's both led and contributed to several interdisciplinary and visionary research programs, including ENSA, InRoot, and the Wheat Alliance. So not only has Simona published in leading scientific journals, but she also holds multiple patents related to plant receptor engineering. And what's really good is Simona has supervised numerous students and postdoctoral researchers and remains strongly committed to fostering inclusive, collaborative and diverse scientific communities. So at this point, I would like to hand over to Simona, where I welcome her to present Architecting Life from Below, Root Control of Plant Nutrition and Resilience through Microbial Partnerships. So hopefully this will work seamlessly, and Simona will be able to share her screen and unmute herself. And just a quick reminder, yeah, if everyone can mute themselves, that would be appreciated.
Thank you very much, Ken. And is that okay? Is everything in place there?
It's good. There we go. Perfect. It's now in presenter mode, so all good.
Good. So thank you very much to all being here, and special thanks to the committee, the ISRR committee, for providing this honor, to giving this honor. I'm really humbled and honored about this award. it both acknowledges the achievements that we together we have done throughout the years, and as well it also obliges us to continue doing high impact and good solid research in the space of the root soil and plant microbe interactions. So I'm really grateful for this award, and I hope that we can all enjoy a fantastic day with fantastic talks. As well, thank you to the colleagues that accepted to talk today, to present their research today. I'm really acknowledged. I would like to acknowledge their willingness to take some time of their busy day and be part of this. And to all those that enjoyed and enrolled for seeing this workshop, which I think it's a very, very good idea. So. The life that we know now on Earth has been definitely shaped by evolution, but we definitely know that it's in the last years it has also been shaped dramatically by us humans. And we have contributed throughout many years to discoveries and improving varieties of different crops that we now cultivate. We improved our capacity to do agriculture. We intensified the agriculture, and this has been key for providing us with enough food to feed the growing population of us on Earth. But this intensive agriculture has been always based on high levels of chemical fertilizers, of pesticides, herbicides, insecticides, and heavy, intense use of soil. And this we know that has led to major disturbances both in the soil, but as well at the global level. Even though we as a population have been thriving, our world, globe has been and it is under tremendous challenge. And I don't want to go too much into these details. We that work on plant science and plant research, we know that there are major challenges that the global level we face regarding the nitrogen and phosphorus and losing the genetic diversity of different crops. So we have a big challenge ahead of us, and how should we tackle these challenges? And it is clear that around the globe, and our research fits into the research that is done throughout the globe by fantastic colleagues around there. And we all work and try to identify both short-term solutions that can help us and work and find solutions for now and here, adapted to these locations and to these environments. But we also know that we need long-term transformational solutions, because we cannot keep on doing agriculture the same way that we have done in the past 50, 60 years. So we need both. And to have the impact that we need at the global level, I think that we need sufficient and enough discovery science, and discovery science that has an aim, it has an impact. And the way we have approached this need for impact and need for discovery science has been to learn from nature, try to understand how nature has evolved and has led to these wonderful interactions between plants and microbes, and try to be mindful engineers of these capacities, engineer new capacities in hosts and plants where these capacities do not exist or are not optimal. We have taken an approach to really look at the discovery science, and we have looked at the nitty-gritty details in the cell, the receptors that sit at the plasma membrane and activate the perceived signals. So we look at these nitty-gritty details, but with the intent of understanding and exploring what I think it is a gold mine under our feet. And we know that there is a great potential for plants to make use of the nitrogen and phosphorus reserves that are around the globe. And iron for other like an example for other metals that are of need, but as well to use these interactions that established under our feet to protect themselves, to protect plants for drought and for pathogens. So I think that this is we are staying now at an edge of really remarkable discoveries and remarkable innovations, both in agriculture, in science, and with the AI that will enable us to make huge leaps in the future and develop these both short-term but as well transformational solutions and identify those. What I think that it is really remarkable when we think about plants is that when they enter the soil and they grow their roots in the soil, they have the capacity to accommodate multiple microbes, multiple guests. Some of them can colonize their roots. Others are actually staying on in between the plant root cells. Others need to be avoided because they are pathogens. So one host needs to accommodate and perceive and understand which microbe comes and when and where and how, if they need to be accommodated or they need actually to start an immune response and avoid their colonization. So it is a remarkable system there present at the root soil interface that plants need and are able to maneuver through these complex interactions, because most of the plants that we see now on Earth, they are not attacked by pathogens and they are colonized by microbes. So that's remarkable. And not only that the roots are colonized by microbes, but the plants are also able to secrete in the environment compounds and chemicals that in the end keep the microbial communities outside of their roots as well under a certain level of the control. So in a way, the plants create these ecosystems and are able to keep these ecosystems in a leash, in a control. And seminal work that David has done some years ago when he was in Cologne in the group of Paul Schulze-Leffert has clearly showed that plants have the capacity to recruit from the soil in their rhizosphere and then further on in their roots to recruit specific microbes for colonization. And there is a selective selection of microbes from the soil into the rhizosphere, into the rhizoplane, and inside the roots. So for a microbe, even if it is beneficial and has beneficial traits for the plant, it is important that it has the capacity to be actually recruited, to be enriched in these compartments that are close to the roots and provide the benefits that they are capable of. So root competence is a key trait for microbes to provide their benefits. And we have looked at commensals that colonize two plant species, the legume Lotus japonicus and Arabidopsis thaliana, grown in the same soil. We collected microbes from those plants and asked the questions: do these different plant species, do they recruit whatever type of microbes we put at them, or is it a particular type of selection? And this allowed us to find that actually there is a preference for commensals to be recruited by their native hosts. And this recruitment is actually dependent on metabolically active root systems. So the active presence and active secretion of plant chemicals from the plant is important for this preferred colonization by the commensals. So But what actually makes a microbe to be able to be colonizing the root was not possible to disentangle at that time. So together with Ronnie de Jonge from University of Utrecht, we embarked on a large study where we actually looked at 3 different plant species, Legume, Lotus japonicus, Arabidopsis thaliana, and barley as a cereal. And we have used large culture collections isolated from these plant species, and we wanted to investigate what are the functions and the traits that make these bacteria able to colonize one or the other hosts. We sequenced all these genomes of these bacterial genomes, we analyzed their functional diversity, and we assembled highly diverse synthetic communities, either derived from these specific hosts or brought together in a large super syncom experiment. We exposed hosts to these 4 different synthetic communities. We looked at what plants actually recruit, independent of the soil. So what do they recruit if there is no soil influence that can have an impact on microbial survival? And we looked at the functions that are important for recruitment. And there are a lot of we had a lot of findings in this study. And so I'm going to share some of those that I think that are really important for our understanding what determines the root competence. One of the finding is that actually when we expose the 3 hosts to the 4 different synthetic communities, we observed that Lotus japonicus, the legume, has been very selective in selecting the same KOs, the same functions over and over again, irrespective of the community that we have applied to those. So Lotus is much more selective in terms of the functions that it actually recruits compared to Arabidopsis and barley. The other finding that we had is that when we looked at the functional diversity associated with these root communities, it was actually the functionality that it is encapsulated, encoded globally at the level of the family that could explain the differences that we found between the root communities in these different associations. So in other words, the diversity of functions that a family of bacteria has when is assembled in these communities it's helping the communities to be differentiated from one host to another or from an inoculum to another. And importantly, when we look within a particular family, it is the isolates that encode the largest diversity of the family itself have the highest capacity to colonize the roots, that have the highest root competence. So, of course, this has one could think that this can have penalties at the level of the genomes, because bacteria cannot increase their genomes just to accommodate more and more functions. It has disadvantages, but these are the observations that we had, that across families, it is the isolates that encompass the largest diversity of this family that have the highest capacity to colonize the roots. And I think that this is important when we think of how do we select these different bacteria to use as bioinoculants, and how they can have the capacity to colonize a particular niche on the roots. And I think that this could be very important for our research moving forward. In addition to these findings, when we look across the 3 hosts inoculated with all these 4 synthetic communities, we have identified that throughout these 12 different combinations, there is a core set of bacterial functions, 266, that are constantly enriched by, irrespective of the host, irrespective of the syncom. So it is a core set, a minimal core set that it has been always enriched. And we looked throughout our communities to see if bacteria that contain these functions will have an advantage when colonizing plant roots. And indeed, this is the case. We found that for particular functions that we are included in these 266, we constantly see that bacteria that contain them, they have a larger abundance in the root compared to those bacteria that do not have this contain these functions. So this is again, these are important traits that we could try to use and seek for in the microbes to be used in the future. Are these functions that we have identified being present in one bacteria? Can it be one bacteria? Can we have one bacteria that has all of them? And at least from the communities that we have looked at, the answer is no. We did not find any that has all these key functions, indicating that the assembly of these core functions is actually a trait that is established at the community level. It's a communal effort to bring in these KOs. And importantly, none of the isolates was completely devoid of all these 266 KOs. So some bacteria, there is a degree of variation between isolates with respect to their capacity to encode these functions. But more importantly, when we compared the results that we had in our studies using synthetic communities with a previous study that has been that has been very well annotated and have looked at different bacteria deposited in the different databases and used different both non-plant associated and soil bacteria as well as plant associated, it was clear to us and we observed that actually these 266 functions that we have identified are actually a response specifically or enriched in the plant-associated microbes. So this is good. It tells us that these are plant-associated functions that microbes have. So as a summary, this large study that we have performed, it told us that different hosts have different strategies for selecting the microbes that they enrich in the root communities. That the communities of lotus, the only legume we had in our study, it is more restrictive in terms of the isolates, the number of isolates as well as the functions that they encode. So in a way, lotus uses a strategy like Swiss Army strategy, fewer knives, but they can do a lot of functions. And then we have identified a core set of functions that provide bacteria with a competitive advantage. So this study has been done on 3 hosts, 3 different hosts. Barley is a crop. Lotus and Arabidopsis are not. So we are taking these learnings now from this plant species, and we take it over to try to understand and study the role of these findings in wheat. We have initiated 2 large programs, one looking at the host genetic diversity and the impact of host genetics on recruiting beneficial microbes in the root. Is a large consortia, and I thank all the partners involved for their commitment to this program. And the other program, it's an interdisciplinary program where we try to understand and how whether it is possible to improve the diazotroph recruitment and association with wheat roots. So it is clear that some microbes have a root competency and are better at colonizing the roots, but there are clear microbes that have identified strategies on how they actually colonize the roots very efficiently, and they have the capacity to be accommodated in the plant cells intracellularly, like the arbuscular mycorrhizal fungi and Rhizobium symbionts. And arbuscular mycorrhizal symbiosis is one of the most ancient symbiosis that plants have developed and helped plants colonizing the earth when they moved from the oceans to the bare rocks. It is a a fantastic association that helps most of the plants here on Earth to acquire nutrients out from the soil. The fungus spreads into the soil and then supplies the host with phosphorus, water, and nitrogen in response to photoassimilates like fatty acids and some sugars. And I'm looking forward to the talk that Uta will have later on, telling us a little bit more about this association between plants and arbuscular mycorrhizal fungi. Later during plant evolution, legumes became very good at accommodating another microbe intracellularly, and that's the nitrogen-fixing bacteria. They even create a special organ, the root nodule, where bacteria is allowed to colonize, to be infected intracellularly, and to amplify. It becomes practically a factory for ammonia production inside the plant roots. And the legumes have become very good at doing this and help them to spread and evolve, evolve different numerous plant species. But this is a trait that it is helpful for legumes, and the other crops that many of them that we currently like to eat and we grow on large surfaces like cereals do not have this capacity. And this is something that the EMSA program is currently doing and trying to improve the associations these crops have, like cassava and maize, to improve the association with the arbuscular mycorrhizal fungi, and as well to engineer these associations with nitrogen-fixing bacteria present in the soil. And this is again as another large collaborative project. It's a consortium, and it's a fantastic work together with the team to try to bring this visionary and these moonshots projects to success. So our strategy in EMSA is actually to use the genetic chassis that arbuscular mycorrhiza is using to be accommodated into the plant cells, to actually adapt these to enable association with rhizobial bacteria. And we do this using on plants that currently are not able to do this, like barley, maize, and cassava. We know that engineering nitrogen-fixing symbiosis is a moonshot, and we have to engineer different stages of this symbiosis. It's a complex program, and what it was important for us is and it is still important for us, is to ensure that now these crops that normally will not recognize the nitrogen-fixing rhizobia as a symbiont will actually be able to do this. And we base this again from learning from nature how legumes are actually able to recognize the symbionts. And we know that when they are nitrogen starved, they will release into the rhizosphere. They lure the microbes, the symbiotic bacteria, into their roots. But these isoflavonoids work as signals for bacteria to initiate the production of the nut factors, the symbiotic signaling molecules, species that are species specific, and they are recognized in the host, in the legume, by lysine receptor kinases, and activate nodule organogenesis and infection threat formation. We know that receptors that recognize these signals are key for symbiosis. If they are absent, legumes are not able to engage and they are nitrogen starved. And now we also know that bringing these two receptors, NFR1 and NFR5, in a complex, in the absence of rhizobia, it is able to initiate the entire symbiotic process, leading to nodular organogenesis first. So this complex, it is the seed for signaling for nitrogen-fixing symbiosis. When we look at these lysine receptor kinases, it is clear that they are inventions of land plants, and they are present throughout plant kingdom. In most of the plants, they are important for recognizing chitin as a immunogenic epitope. They are important as well whenever they have been studied in emerged. They are important for the AM symbiosis, and studies in Uta's group also show that they are important for lateral root organogenesis. They have lysine domains as exposed to the out of the plasma membrane, a transmembrane, and intracellular regions that contain a kinase domains. So these are present throughout plant kingdom, but when we look at the legumes, it is clear they have evolved these NOT factor receptors that are very similar to the chitin, to immune receptors, and now they have the capacity to enable legumes to activate root nodule symbiosis. So what is different between the chitin receptors and NOT factor receptors? It emerged clearly when in Giles Oldroyd's group they have been comparing the capacity of barley and medicago to respond to this type of signaling molecules, carbohydrate type of molecules, chitin and different lipid oligosaccharides, NOT factor types. So it was clear that barley can perceive all of these type of molecules and respond similarly with the same sensitivity to these signaling molecules, while medicago, the legume, had a much more differentiated type of response that most of the non-symbiotic or non-compatible type of carbohydrate signals induced a response that was more or less similar to the one in barley, but the response of the legume to the compatible NOT factor was much more sensitive, so around 1,000 times fold more sensitive. So these receptors provide the legume plant with both increased sensitivity to the NOT factors as well as increased specificity, because using NOT factors that are not compatible with medicago was not sensitive enough. So this told us that we need to engineer in these receptors both the specificity and the sensitivity. So our strategy was to compare the receptors that activate the immunity and their similar partners that activate the symbiosis, and then use the learnings from these comparisons and functional analysis, use these learnings to afterwards engineer in the target crops. So when we looked at these comparisons between the immune receptors versus the symbiotic receptors, it was clear to us that we need to understand both how the ligand perception takes place, which regions, which domains are actually important for recognizing chitin versus NOT factors, but as well it was important to identify how the intracellular regions have the capacity, after recognizing the specific ligand, to activate actually immunity or symbiosis, because in legumes these type of receptors are expressed in the same cells. So this told us that the specificity is encoded at the level of the receptors. So we embarked in these studies, and first we looked at the ectodomains of these receptors, and we have identified that in legumes, both chitin and NOT factor receptors have in the lysine 1 domain specific regions, we call them region 2 and region 4, that are actually important for recognizing the ligand. And this is both for the chitin and as well as for the NOT factor perception. So these two regions are important and are required and are sufficient for recognition of LCOs in the legumes. In the group of Kasper Andersen, the structures for these ectodomains have been obtained, and when we looked at where these regions are actually present, we observed that for chitin receptors, these regions throughout legumes are highly conserved, and this makes sense because chitin it has an invariable structure, so it is important that there are not many variations between legume species when it comes to recognizing chitin. However, when we looked at the NOT factor receptors, there was the opposite situation, that these regions were actually highly variable. And this again makes sense because the knot factors that are produced by different rhizobia have different structure. They do have different decorations on the chitin backbone. So these decorations need to be accommodated by these regions present on the ectodomain of the NFR1. And we have been able to show that it is actually possible to engineer knot factor recognition in the chitin receptor ectodomain, and this was sufficient to activate knot factor binding, but as well the functionality and restore the nodule formation in the legumes. When it comes to the intracellular signaling of the NFR1, it was clear to us that it is if we have the ectodomain that recognizes the knot factor, it is also important to take into consideration what parts of the intracellular region are important to activate the symbiosis and not the immunity. So we have then looked at these intracellular regions, and we have identified that there is a specific region sitting just before the kinase region of NFR1 that contains what we call a symbiosis determinant. And this symbiosis determinant, it is very similar between the chitin and the immune receptor and the symbiotic one. There are only 6 amino acids residues that differ between these two receptors. However, the symbiotic one is the only form of this, is the one able to activate root nodule symbiosis. And we have been able to show that actually out of these 6 residues, 2 are key for activating root nodule symbiosis. And it was sufficient to exchange these 2 residues in the chitin version of the intracellular region to the one that present in the knot factor receptor and ensure root nodule formation in these roots. We showed this that it is possible with the chitin receptor from lotus, but as well using a receptor from barley, now equipped with the capacity to recognize the knot factor, and just with this symbiotic determinant from the knot factor receptor, it was now the barley receptor was able to activate root nodule symbiosis in the legume. So this is really exciting. That tells us that with minimal changes, we can actually re-engineer the signaling pathway that is activated in these plant roots. What I think is also a very important finding from our studies was actually that it is important to have a kinase, an intracellular region of these receptors that can drive both immunity or symbiosis, depending on both types, depending on what kind of ectodomain do we have. So if we provide the intracellular region that has the N-terminal part from the immune receptor and the C-terminus from the symbiotic receptor, and provide this form of the intracellular part with the ectodomain for recognizing the knot factor, then this will activate the symbiosis. The same version of the intracellular part, but now with the ectodomain recognizing chitin, will work in immunity. So this tells us that it is possible, this evolution of this specificity has specificity for the knot factor symbiosis has not been done at the cost of the immunity. It has evolved in different regions of the intracellular region of the receptor. So for the knot factor receptor 1, we think that we have key understandings for how to engineer the ectodomains as well as the intracellular regions to activate the root nodule symbiosis. When it comes to the knot factor receptor 5, the partner of NFR1, together, as I mentioned, they are important and sufficient to activate root nodule symbiosis. We have also been able to identify that the ectodomain of the NFR5 is the one responsible for deciphering between chitin and knot factor, and it was possible we have found a hydrophobic patch in the lysine 2 domain that it is responsible for this capacity. As well as for the intracellular part of the NFR5, we have identified as well a region in the juxtamembrane that it is important for symbiotic signaling. So we have exciting findings, and I think we are really now at the stage where we can take these findings and move it into the target crops, into the cereals. And there is exciting science coming from this engineering now in maize and barley receptors, NFR5 type of receptors. And this is just a teaser to tell you, keep an eye on this. There is going to be exciting things coming up hopefully in the near future. So it's really an exciting time here now. So we have identified these critical novelties that evolution has led to the emergence of the NOT factor receptors, as well as maintaining the tithing immunity at the same time. We know that new ligand binding domains have evolved in NFR1, that we have new binding specificity and new binding mode in the lysine 2 of the NFR5. And then when it comes to the intracellular signaling, we have identified both this symbiotic determinant in the juxta membrane of NFR1, and that it is important for the NFR1 to lose the capacity to activate the immunity. Otherwise, we get a much more reduced symbiotic activity. And importantly, that these two receptors, they are the key for activating the symbiotic signaling in legumes. So yes, we do work at the nitty-gritty details, and we try to understand how these molecules are actually able to activate a specific signaling in the plant cells. But what I think it is important to remember, and that's something that always brings me, it's very important to understand the mechanism that ensures one particular signaling pathway. But we need to understand as well that a plant as a host is able to accommodate these different signaling pathways within the same cell. How is the plant actually able to recognize that this particular cell is now encountering an arbuscular mycorrhizal fungi, and it has to accommodate in this particular niche the arbuscular mycorrhizal fungi, while in the other region of the root will accommodate the rhizobia? It's still a question for us, and I think it's really a fundamental question now to understand how is the plant regulating at the entire organism level these different associations with these different microbes. And I think we in the plant science, we have a fantastic opportunity to actually address this and disentangle this signaling at the cellular level, because this is not possible to study in the animal gut. They will never be able to disentangle this at the cellular level. At least that's what I think. So I think it's a fantastic opportunity for us in the plant science to embark on these studies. And again, how these actually lead to larger influences. We know that symbiosis in legumes with nitrogen-fixing bacteria, and the signaling that it is exchange between these the two partners is not only important for nitrogen-fixing symbiosis. We have shown that it is also important for accommodating the larger microbial, modulating the larger microbial communities surrounding the legume roots. So it is an active response and an active remodeling of the exudates and the metabolites that are sent out into the rhizosphere that plays a major role in this root competency and maintenance in the root soil interface. And I think we are able now to mine this gold mine and to understand how we can use these relationships that establish between microbes and the plants in the underground to actually enable us to use them and engineer them with a mindful exercise, because we have to learn from what has been done during this intensivization of the agriculture, that everything that we do, we have a responsibility to bring up to life technologies that are viable and are sustainable on the long term, with the long-term mission. So with this, I'm going to end up. And thank you all the members and the close collaborators that I had throughout these years. All of this that I presented, it is the result of your work. I'm just here, trying to guide and help and mediate and inspire, I hope. So thank you all for contributing to all of this. Special thanks to colleagues at our section, at the plant section. It's fantastic to have wonderful colleagues that we have such good interactions and complementary expertises that enable us to go beyond the trivial let's say trivial questions, and dare more. All colleagues at the department and colleagues around the world, I will have to say that without fantastic collaborations and fantastic team, we would not have been able to reach so far. So that's it is collaboration and diversifying and taking advantage of the expertise that exists throughout the world. This is what it makes us, at least makes me waking up this morning and come back here and tell you what we want to do and where do we think that we can go. And thank you all online. It's I hope that this inspired some or sparked some questions, and thank you for being here. Thank you for listening.
Thank you very much, Simona. A very, very interesting talk there. Yeah, very fascinating. Just going round, do we have any questions from people? So yeah, there's one in chat here from Bhupinder Singh. So I'm interested to know about the epiphytic mycorrhizae that can be used for abiotic stress tolerance, particularly heavy metal immobilization in the soil. So do you want to make any comment on that?
Yeah, that's so. I humbly have to say that we have been focusing throughout our research on these interactions that are very well studied at the genetic level, like arbuscular mycorrhizal symbiosis, and not that much, but as well the rhizobial symbiosis. But the diversity of interactions that exist throughout the soils and the endophytic associations with both the bacteria and fungi are. I totally think that they are unexplored, and how they can contribute to plant resilience and response to stresses. This is fascinating, and I think we should be able to study this and should study this in more detail. Unfortunately, I don't have, from our research, we don't have any details regarding this. But I'm pretty sure that there is fascinating research throughout the globe on this aspect. So yes, watch the space.
Thank you. Do we have any more questions from the audience? You're more than welcome to raise a hand. So I appreciate there are some people with you in person. Simona, do you have any questions from the audience with you?
Any questions?
Yeah, I see one at the front right there.
Thank you very much. Really great talk and overview of interactions and which is kind of the booming field. It's really exciting. But yeah, it's has a great potential for the users of that. But how do you think, what is a major challenge or limiting factor preventing translation to research, and when will you overcome that?
Yeah. Oh, that's a challenging question as well. Very, very good question. It is true that it is. We have high expectations from the plant microbiome studies and what it has the potential to deliver. And so far, I think we are still at the level of the discovery. If we think about how many years of research in the root nodule symbiosis, of genetics of root nodule symbiosis, have been needed until we started thinking about the possibility of engineering and moving this into impact. The amount of research and the time that has been so far in the microbiome, it's still in the incipient stage, I would say. So I think, first of all, I think what we need is patience, maybe, and the understanding that we need to still to do more discovery science in the field. The second thing is that it is a highly complex community here. We are dealing microbes that are so diverse, and we need to understand this complexity and to be able to understand it, but as well to model it and to predict afterwards. And I think there is a major. I think that it's a great potential from taking the learnings from the lab, from, let's say, more reduced or different levels of synthetic communities like we have used, others have used with reduced diversity or larger diversity, and take these learnings and extrapolate and put them make use of this AI modeling and use the AI technology to help us understanding and predicting I think that's where there is a lot of hope. And I think as well, if we look throughout the research and the plant microbiome research, there is a lot of data there, but it is so much of this data that it is not well annotated. We need to have a good understanding of the data if we want to use it in the larger perspective. And studies that are well designed to answer particular questions, and larger studies, not only, a few synthetic communities like we have done. So I think we need more and very well designed studies for this. That's my opinion. Thank you.
Thank you very much. I'm just going to switch online. Corné, would you like to ask your question, please? Feel free to unmute.
Yeah, okay, thank you. Well, Simona, congratulations, and also a very nice talk. It's a nice example of how you can nicely integrate research on rhizobia and on the microbiome, really nice. I have a more philosophical question. So you demonstrate that the rhizobia and the nodule formation can make use of, let's say, microbiota in the environment to perform even better. And of course, not much is known about which functions are there needed, but from the perspective of the rhizobium, what do you think that the microbiota can deliver to the rhizobium or the symbiosis so that it can perform better?
Yeah, thank you, Corné. That's a very good question, and I think it's a very good question because the rhizobium inoculants have been the first ones to be taken out to the market, and still I think the ones that are most used throughout the globe, and still the ones that also have challenges, because we know in a particular inoculum might work in this location or this year, but then in a different location might be a completely different story. And I think that one of the findings that we had from our super syncom experiment might provide some explanations for why this happens, because now we know, at least our findings tells us that within a family, bacterial family, the isolates that have the highest encompass the largest diversity of that family in that particular soil will have the largest competence. At least this is what we have seen. So now if we extrapolate this to the rhizobial inoculants, we have a particular isolate. It might be that in a particular soil, meeting the functions that the rhizobia family has, that particular isolate is doing well because it covers this diversity. But in a different soil, the rhizobial diversity is much larger or different, and then the inoculant that we are using is now it has a disadvantage. It cannot compete with the rest. And I think the competition that exists between the inoculant that we use and the native population that exists in the soil is key for us for understanding how we can use these symbiotic associations to the maximum potential, because we do have fantastic inoculants, that do very well in the lab or in the greenhouse in controlled conditions. But when we put them out into the soil, it's a different problem there.
Yeah, okay. Well, thank you.
Thank you. Just a question from Davide. So will you look into barley genetic resources, e.g. RLK4, within wild and domestic genotypes, to identify variants which may be more prone or less prone to accommodate symbionts?
Yeah, that's a very good question, David. Yes, there is a large diversity of barley RLK4 there. What it has been shown, not in barley, and we didn't explore this maybe to its full potential, but it has been shown, for example, in rice. There is a study in rice that showed that genetic diversity within SERC receptor, that would be the RLK4 for barley, actually led to the identification of varieties that have a better mycorrhizal association compared to the other varieties. So it's clearly a very good scope in looking into the genetic diversity that exists in the natural populations. That's a very good point. Yes.
Thank you. Just another couple of questions in the chat. So Clara said, could you explain the figure on one of the last slides where it said nitrate in soil symbiosis non-permitted and symbiosis permissive on the right?
Yeah, I sort of use that more like a concept figure without putting the details onto this. So this figure, it comes from our studies in Lotus japonicus, when we have the legume, where we have tried to understand how is what is the impact of nitrogen nutrition from the soil versus nitrogen nutrition provided by the symbiont. And there we have found that in the presence of the nitrogen in the soil, so nitrate in the soil, the microbial communities were still diverse, but the connectivity that existed within the root communities was much lower compared to what we have observed when we have the symbiotic permissive condition, where again the diversity was larger. There were much more connectivity, and as well the communities were changed by the presence of a symbiotic active rhizobia. So we believe that both the different types of nutritions create different states in the plant, and the plant responds to this either nitrogen provided or symbiotic nitrogen provided, responds differently, and in the end responds differently with different exudates that we have shown that there are different exudate patterns in these conditions, and this leads to we think that this can lead to different associations at the root soil interface. This is a work that we have published in last year in 2024.
Thank you. And then a final question from Olivier. So can maize or barley be transformed so that infection threads are formed?
Yeah, that's a very good question. I think infection by rhizobia is one of the key challenges for engineering nitrogen fixing symbiosis in cereals. We will see. It's whether the infection needs to look like an infection thread or exactly how it looks in legumes, where it has evolved to be very well specialized and very well guided. There are a lot of controls during the infection process, or we can actually engineer a version of this infection. It's a very good question, but we'll see. It's a moonshot challenge, but there is great progress in this understanding in the ENSA program. So yeah, we are definitely optimistic on this.
So now on to the official award for the Dundee Root Medal. Now, I believe there should be someone with you in our house to make the presentation. So if you wish to spin the camera around so we can see the official award, and maybe hear a few words from the presenter if they wish.
So, Simona, it's a great pleasure and honor for me to present you this prestigious Dundee Root Medal. I'm delighted on behalf of the people that you have worked with, and also the Department of Molecular Biology and Genetics here at Oxford University, to really congratulate you with this achievement. I think really this highlights your several contributions to science over the years, and I'm very happy to see that you receive this. So here we go.
Thank you very much. Thank you very much, everyone. It's a great honor, and thank you to the committee for choosing me for this annual award. And thank you. I hope that we can meet. I hope that I can get a list of participants, and whenever we meet at some conferences or abroad, that we can have a glass of champagne or or something to celebrate. Thank you. Thank you.
No problem, and thank you very much, Simona, for your contributions before today, and also for your talk that you've given. So thank you very much.
Right, thanks, Ken. Thanks for introducing the session as well. So we'll be joining you here from Napier, Bilbao in Spain. Not as good as sunny Dundee, which we wish all we were there, but we'll try to have this session run as smoothly as possible. So yeah, so our first speaker for this session is William King, so expert in microbial ecology. And he's going to talk about a topic that is quite important and exciting at the moment, which is how soil microbiome can contribute to improve plant fitness and stress. So William, on to you if you want to get started. So I cannot advise how to share or to do things.
Oh, good. We did a test run this morning, so hopefully this works.
Excellent.
And does that look good for you guys?
Yep, for me.
Perfect. Excellent. Okay, so first of all, thank you for the invitation to present. Today I'm going to be talking about how manipulating below ground feedback between plants and microbes to alter how plants respond or perceive stress. And so why drought? So with climate change, we're expecting quite large changes in precipitation and rainfall patterns around the planet. And we're expecting to see changes in the severity and duration of droughts. This is further compounded with a growing population, which obviously needs to be fed, so putting constraints on available space to grow crops, but then also demands on actual freshwater usage. So we're expecting freshwater demand to double in the next couple of decades. And so I always like to put up this little quote here, which says, drought alone causes more annual loss in crop yield than all pathogens combined. So it's quite a significant problem that we need to deal with. Oh, what happened? Hold on. There we go. And so quite a lot of research groups around the world are looking at ways that we can mitigate drought or find ways to deal with drought. And there are quite a lot of ways to deal with it. And so you have a lot of the traditional approaches, for example, traditional breeding, so looking at ways to manipulate water use efficiency, the bioengineering approaches, so genetic editing and what have you. And then, with these approaches, trying to alter how plants architecture, root architecture, morphology, how they deal with water uptake. But then we also have stimulants as well. So this is more from the chemical side. So adding physical chemicals to the plants to alter how they sort of grow or how they perceive the environment. But what my group is quite interested in is this type of feedback between plants and microbes. So manipulating the crosstalk between hosts and microbes to alter how plants perceive stress and deal with stress. And so for the rest of my talk, I will be talking about managing microorganisms and their functions. So why manage microorganisms? You're probably sitting there going, well, this is such a silly thing. What are you talking about? If you take any soil on the planet, even from the bottom of your shoe, you will find plenty of microbes that can do all of the functions that we need. For example, nitrogen fixation, phosphorus solubilization, contaminant degradation, plant growth promoters, they're everywhere. Why exactly do we need to manage these things, particularly in agricultural settings? And the main factor is that just because these functions are present, it doesn't necessarily mean that they are optimized. And so here I have three different functions, if you will, and their sort of efficiency. So you can see that, with these particular functions I pulled out, there is quite a range of efficiency or how well microbes can do these particular functions. So there's quite a degree of optimization that can occur. And so this idea of managing microorganisms, it's not new. I didn't come up with it myself, and Simona actually mentioned nitrogen-fixing inoculants in her talk. This idea has been around for over 100 years now. This is one of the first inoculants that was developed. But there's been quite an interest in recent times, particularly by agricultural biotech companies, in trying to manipulate microbes by producing these microbial inoculants to sort of introduce microbes into soils to augment functions of interest. Now, for the most part, these inoculants do work, but they are held back by unpredictable establishment. And so there's this lovely study here by Hidri back in 2016, where they found that the majority of the time, inoculants seem to have a positive effect on crop yields, but there was quite a big chunk where they didn't do anything. And there was actually a small chunk where it was negative. So you had negative effects on yield. So it is quite unpredictable. And this is because these inoculants, well, at least what I think, these inoculants have to go through quite a lot of challenges. So for example, we have the abiotic constraint. So this is the physical environment itself. So you're introducing a bacteria or fungus or an inoculant from a different environment into a non-native soil. And so it has to deal with changes in pH, oxygen content, water content, nutrient content. So it's a mismatch in their physical environment. Then we have the biotic constraints. So these inoculants have to actually integrate into an existing community, talk to other members of these communities, outcompete them, actually establish in these communities themselves. And what industry is really, really good at is they can pick out a soil, screen for any function that they're looking for, like thousands and thousands of microbes, for example, nitrogen fixation or phosphorus solubilization, and then they're really good at scaling that up to make an inoculant. The problem with this particular process is that it does not capture the ecology of the system. So it's really just to pull these microbes out and grow them up really quick. And so we're missing that aspect. And so that's where this idea of sort of whole microbiome selection comes in, where you take a whole microbiome and you subject it to a particular stress or a particular selective pressure, if you will, of interest. So for example, drought. And so what that does is that essentially conditions those microbes to, or filters those microbes for the ones that deal with drought the best. And then you end up with sort of a refined community, if you will. And this type of approach has a few different benefits, like retaining species interactions, maintaining metabolic networks, improving microbial establishment. So we know this as well. And also, this is not something that I just came up with on the spot. We do have quite a lot of evidence from the literature that this type of selection can be quite beneficial, and it does work. So for example, we know that by selecting a microbiome, you can improve plant biomass. We know you can alter plant flowering time with soil microorganisms, and the microbial diversity in a soil, we know that can actually impact the above ground plant diversity that can actually exist in that environment as well. And we've done this before in the past as well by depleting and reintroducing functions of interest like nitrification. And so this sort of begs the question here of why not use a whole microbiome? And really the type of thing that we are manipulating here is simply environmental filtering, where essentially you take a whole community, you put it under a filter or a selective pressure, and then the subset of those that can deal with that well makes it through. And then you go again through another selective pressure, another filter, and it just sort of goes through and filters the community for the subset that deal with that pressure the best. Another way you can look at this as well is the root microbiome. Simona had a nice schematic of this already from Dundee, which is fantastic. So essentially you just filter through from the bulk soil microbiome to the ones that can deal with tolerating the root, then the root environment, and then the endosphere. So it's this type of environmental filtering that we're manipulating here. And so in this particular project, what we wanted to do was use drought as our sort of filter or selective pressure to condition or passage a microbiome And so we had two hypotheses going into this. The first one was by providing drought-adapted soil microbiomes to plants, it would improve their plant fitness under drought. Makes sense, right? Give them drought-associated microbes. The plant should do better under drought. And then in the other instance, it is providing wet-adapted soil microbiomes to plants will decrease the plant's fitness under drought. And so this was our experimental design. Now this is a lot to show you on the screen, so I'm going to walk you through it. So for the first part of this experiment, what I did was I went out and collected 4 different soils. So it was 2 agricultural soils, an urban soil, and a forest soil. And I used that as my sort of starting microbial pool. So I wanted a diverse starting set of microbes. I then used that soil to grow with maize, and that was the maize microbial pool. So we enriched for sort of maize-associated microbes here. Then this maize microbial pool got split off into 2 directions. The first was this add-back microbiome control. So some of the soil was frozen to use as an add-back microbiome control. And this acts as our reference point. So every sort of generation of selection, or anytime we do things with plants, we have this control so we can actually reference back to something. And this really accounts for things like greenhouse effects and, things like that. The part of the maize microbial pool that the rest of the maize microbial pool underwent this sort of conditioning experiment, where for 4 weeks, they were either droughted, so they didn't get touched by any water, completely dry, or it was kept completely wet, so they never ever dried out. And then every time I did this type of conditioning, at the end of the 4 weeks, I would have a plant experiment. So we used an inoculum, a 10% inoculum, and then we gave that to maize, and then put the maize under drought stress. So anytime you see any type of plant data, it's droughted plants. At the same time, I then also use this sort of conditioning experiment as an inoculum for another round of selection. So we do multiple rounds of this type of selection, or what I call generations of selection. And again, every generation we have these plants here. So for all intents and purposes, the only difference between these treatments are the physical microorganisms themselves that differ. At some point, we also looked at the culturome. So we did some high throughput culturing to pull out some bacteria associated with these plants as well. Then at the very end of this experiment, we had this transplant sort of crossover experiment where we did the same thing. So this is that soil I was talking about. This is the conditioned soil, and that underwent drought conditions, but we also watered some of those plants. And so here we did drought and wet conditions, and I also took the rhizosphere soil from the plants in the previous generation and carried that forward. So we had this 2 different types of soil inoculum, and then they were droughted and they were wet. Right now, I'm going to talk about this starting part here, and then we'll come back to the other part in a second. So what we saw was that iterative drought selection improved plant fitness. So what we're looking at here, this is the drought associated or the drought conditioned microbiome relative to that add back control that I was telling you about. And then we have 3 different measures here. We have above ground biomass relative to the microbiome control, then below ground biomass, and then stomatal conductance. And then on the x-axis, we have generation. So you have 1 through to 7, and then 1 through to 6. I just forgot to do 7 here, but it's the same pattern. And what you can see is that with more rounds of selection, you can see that the plants progressively deal with drought better over time. So the only differences here are the microbes that I provide these plants themselves. So they are progressively getting better. On the flip side, we saw with the watered or the continually wet adapted microbiome, those plants did worse over time. So they progressively got worse with the above ground biomass, below ground biomass, and the photosynthetic rate as well, which matches what we were thinking. When we look at the microbiomes of these plants, these are the rhizosphere, and this particular one is for bacteria. We're looking at an ordination here, and the way it works is that each of these different colors are a different generation of selection, with the red ones being the first generation and sort of this magenta, purpley thing being the final generation of selection. And I'll pick out the sort of patterns of interest here. But here we have the add back control, and you can see that they cluster quite nicely together. There's a bit of movement, but everything's sort of together, which is good because that's our reference point. For the drought microbiomes, or the drought adapted microbiomes, you can see that we see this movement up the top here, starting from generation 1 all the way up to 7 at the top. And then on the wet side, it's over here. So quite a significant or severe selection here, as you can see. So starting from down the bottom and moving up to this corner here. I was quite curious looking at this pattern to see how strong that type of selection was, if we could quantify that. And so what we're looking at here is Bray-Curtis dissimilarities relative to the add-back control in each generation. And the way this graph works is anything higher on the y-axis here means it's more dissimilar. And so anything down here is more similar. And you can see, I just want to point out the drought one and the wet one. So the drought one is the orange one here. So quite strong selection. But then you can see on the wet one, this is the wet microbiome, they are very strong. And they ended up being the strongest divergence. It diverged the most from the add-back control, the wet treatment. When we look at fungi, it's not as clear-cut. So we don't quite see this type of generation to generation movement. But what we did see was that it tended to cluster based on treatment. So whatever treatment you pick for the fungi, it sort of sticks in that one, and that's what it stays at. So we have our add-back dry here in the red. I know the colors are confusing. I'm just thinking about that now. And then we've got the droughted ones up the top here and the wet ones over the side here. In terms of responses of the actual plant roots themselves, what we're looking at here is specific root length of the seminal roots relative to the microbiome control. And what we saw was that the specific root lengths of plants, droughted plants, given the droughted microbiome, they increase over time, whereas for the wet adapted microbiome, they flatline. So there's no difference there. So we're seeing a difference in the seminal roots. They're responding to the microbiomes we're giving them. For the culturome itself, I mentioned that we did some high throughput culturing. This is using a fancy device called the Galt Prospector, which we had at Penn State University, which I think Cornell has one. Actually, it's a pretty nifty machine. We did see shifts in the culturome as well. And so for the sort of wet microbiomes here, they start off here, and after a couple of generations of selection, they move over here. And then for the droughted ones, we see that they move from over here to over here. So we are seeing this fingerprint come through with the cultural component as well as the culture independent component. For the crossover experiment with the conditioned soil and the rhizosphere soil, I'm going to focus on the conditioned soil first. So this is exactly what we've just been talking about. What we saw was that the fitness benefits are being conferred or persisting under watered conditions. So what you're looking at here is this is drought conditions on this side here, and this is the watered conditions on this side here. And what we're looking at is the droughted microbiomes with the droughted plants and the wet adapted microbiomes with the droughted plants. And this is the exact same data I've showed you. It's the same pattern. I've just pulled it out into a box plot. So you can see droughted microbiomes help the droughted plants. But when you water these plants, you can see that again, whatever these droughted microbiomes are doing to these plants, it is persisting. So those plants are growing way better under those watered conditions. And we can see that with this is above ground biomass, but then also measured with physiology. So this is synthetic rate down the bottom here. So this is the bacterial component of the microbiome, what you can see here. And so it's an ordination again. And what we saw was that the legacy of what we did to those microbiomes was quite important. So here we have the droughted soil on the left-hand side. So these are the drought-adapted microbiomes. And here we have the wet-adapted microbiomes. And you can see that they cluster based on where they came from. And then you can see that by watering the plants, there was a bit of a shift here, a bit of a host selection shift here. But it still maintained that legacy of where they came from. Now, I mentioned the rhizosphere component as well. This is where things get a little bit interesting, and it's a bit up to interpretation. What we sort of see is that having the host selection seems to alter the patterns a little bit. So what we're looking at here, and again, I appreciate that the box plot colors don't match everything else that I've talked about, so I put these cute little boxes here. Here we have the soil conditioning, so the microbiome conditioning experiment. And then here is the rhizosphere from the previous generation that was passed forward. And this data here is exactly what I've shown you already. Drought microbiome under droughted conditions does better than the wet microbiome under droughted conditions. But when you transfer forward those rhizospheres, what you see is that for the droughted microbiome, they get worse. Okay, so by having some type of host selection, it seems to possibly remove the positive influence of, or at least maybe the host is not selecting for certain microbes in the soil, and therefore we're losing those benefits. And then on the other side, we have the wet adapted microbiome down the bottom here, and then the rhizosphere here. And you can see by having that one generation of selection, you're getting a benefit for the plant under the drought conditions. So to me, that sounds to be possibly that under normal or watered conditions, having that sort of host selection is better. Whereas under sort of droughted conditions, that host selection does not seem to help you. That's my interpretation, at least. In terms of the microbiome, again, this is the bacterial component of the microbiome. You can see that this is very similar to what we're looking at before. You have the droughted soils together here and the wet soils together here, but then the rhizospheres, so this is the transferred forward rhizospheres, they are a little bit different to the soil. So you can see that host fingerprint moving away those microbiomes. When we look at the fungal component of the microbiome, again, we get this broad sort of drought and wet clustering, but it's not quite as clear cut. There is a bit of crossover between the rhizosphere and the droughted soil, possibly linked to fungal lifestyles and how they grow through a soil. So next up, I've managed to pull out quite a lot of isolates from this experiment. So the plan is to go through, figure out what we've got, and then try to do some experiments where we add these back to plants and see how those plants, try to mechanistically figure out what's going on with this crosstalk between the plant and the microbes. And just the take-home message so far. So what we can see is that we can manipulate a whole soil microbiome to alter how plants respond to stress, and they get better over time. You can see that this type of conditioning is quite a strong selective pressure on the microbiome, and so you see quite a strong shift in the microbiome. And we know that these sort of benefits that the drought microbiome provides to the droughted plants, they persist under watered conditions, and there seems to be some type of host effect that maybe decouples this pattern depending on whether it's a drought or watered condition. And with that, I would love to acknowledge all the people that helped in this experiment. Big team at Cornell that helped quite a lot with this, and then also my old advisor, my two old advisors, Taryn Barreley at Cornell and Terry Bell, who's now at the University of Toronto, who helped quite a lot with this experiment itself. And yeah, thank you. With that, I'll take some questions.
Right, thank you very much, William. So maybe Ken, if you can help me with the audience, I don't know how you want to split. So there are, I think, a couple of questions in the chat. So maybe I can start with one question from the chat, which is a long one. So I'm just going to summarize it a little bit. So I think the microbiome might have effect on root, but also on leaves. And the question is, do you have an idea of the mechanisms and the molecular regulatory mechanisms that might affect root and also the shoots?
Yeah, so I suspect it has to just do with hormonal manipulation, whether that's ethylene or some type of ABA signaling or something along those lines, because that would explain how at least we're seeing the differences in the stomatal conductance and photosynthetic rates. So I suspect it has to do with hormonal manipulation, and hopefully when we pull these isolates out, we'll be able to get into that sort of nitty-gritty detail to figure out if that's what it is.
Okay, thanks. So there's another question from Sophia, and is asking, like, during the filtration process, the microbiome filtration, don't you expect some reduction in the community resilience as a result of that? And if you could develop a bit on that.
Yeah, I suspect that, I mean, this is quite a strong filtering, right? So you would have quite a lot of microbes being pulled out. So the resilience of the community should be altered. Whether it is a strong negative effect on resilience, I'm not sure. It potentially could be, but under natural settings, you would get natural dispersal, right? So you do get quite a lot of immigration from surrounding settings, and so you'd expect the resilience to bounce back. Under a controlled setting like a greenhouse, I suspect that would be quite limited, but in a natural setting, that should bounce back. But who knows?
Okay, thank you. So Ken, do you have a question? Maybe you can ask your question directly and maybe check on the audience, and then after we should move on, I think.
Yeah, thanks, Will. Really interesting. I'm just curious about how quickly the microbiome reverts back to initial type after you've done the adjustment to it. I'm just interested about how, what the response time is.
Sure. So I think for this one, we didn't check how quickly it goes back. And I think because of the way we do the filtering and how controlled it is, there is none of this dispersal coming across. So it's kind of hard to measure how it bounces back, because every time we filter, we're essentially removing microbes completely. They're going extinct, right? So in that sense, I wouldn't expect them to just bounce back from some type of dormant microbial pool. They would be gone. But I think in a natural setting, they should bounce back because the microbial dormant seed bank is quite high. Well, for the time frame of that, I couldn't tell you. Yeah.
Okay. Thank you very much. There's one other question from Tasnim. So, is there any microbe that can work well both in drought and wet conditions?
Ugh. I mean, a lot of microbes persist in both conditions, right? If you think about the soil environment, how heterogeneous it is, you do have these fluxes in water and oxygen conditions. So I suspect there must be something that I think most things would deal well with both, but the extremes of each, that I don't know what you'd pull out there. There must be something that deals with it, right?
Yeah. And just before we move on, I appear to have taken over from Lionel. Apologies, Lionel.
Yeah, no, that's fine.
Simona, as you're the medalist, you can ask your question in person. It's quite an interesting one in the chat.
Yeah, very nice job, very nice work, William. I was wondering, do you have any speculations of why do you see this response on the soil versus the rhizosphere, and what could be the where if one should go and try to identify causes, where would you go to look for that?
Yeah, thanks, Simona. It's something I thought about for a long time, and I don't necessarily have a fantastic answer for it. I do know from the literature that when you do this type of drought conditioning on a soil, the benefits that are conferred to a plant host seem to be inadvertent. So it seems to be whatever the plants are doing are trying to survive them, whatever the microbes are doing, they're trying to themselves survive, and that then persists to the plant getting a better, I guess, fitness under drought. The host selection, I'm not too sure. I mean, we do know that exudation changes under drought, abundance and composition does change. So I don't know if there's some type of signal that for modern crops, at least, it's just selecting for particular microbes that either don't really do well under drought. So they're picking these microbes out and then they just die when they get challenged. But there definitely has to be some type of host element. And I know, at least with this type of manipulation, in the literature, most people do host to host transfer, so it's rhizosphere to rhizosphere, and this type of soil conditioning is not as well sort of researched or studied. So I think we really do need to look a bit more in depth about why that's different under drought, at least. Yeah, but good question, Simona. Thank you.
Right, thanks. So I think we're going to move on to the next speaker. I'm not sure I need to introduce Davide Bulgari to you from the University of Dundee. As I'm sure that even if you're remotely interested in rhizosphere microbiome, you'll have heard of his work. So today we'll speak about structure, function, and host control in the rhizosphere. So Davide, if you want to get started.
Okay, so can you hear me?
Yes.
Fantastic. So thank you very much, Lionel, for this very kind introduction. I'm really chuffed to be here with this terrific lineup of speakers to celebrate Simona's achievements. And therefore, I'm thankful for the opportunity given me by the local organizing committee, of which I'm part of. So let's move on. What I would like to do with you in the next 20 minutes or so is providing a very brief introduction on the microbiota at the root-soil interface, prior delving into a series of experiments that we have been carried out since a few years now to try to identify what are the genetic bases regulating plant microbe interactions in croplands. And then I will close up with a couple of slides highlighting where basically we would like to go and the potential for impact in basic science and translational application alike. So we have been doing what we have been doing because we believe that through soil and the interaction between plants and microbes in soil passes global food security. And of course, if you could understand this interaction better, we may be well positioned in making or increase the benefit for mankind. So these are biological processes, and like any biological processes, should be seen through the lenses of evolution. And as alluded by Simona in her talk, what happens to crops, so the hallmark of crop evolution are the processes of domestication and breeding selection that progressively decouple, let's say, modern cultivated varieties from their wild ancestor, and that left a footprint on the capacity of plants in interacting with the soil biota. In particular, modern varieties or elite varieties have been selected to respond to modification with non-renewable inputs in the soil and in the interface between roots and soil. And obviously, the question is whether if we could look back, at the microbiota thriving in association with roots of wild ancestor that thrives in, let's say, more pristine soil, or anyway, they have not evolved to respond to these external inputs, we may find something that could be useful to sustainably enhance crop production nowadays. And because these processes has left a mark in the genetics of the plants, there also the question whether there is an impact on the microbiota that could be traced and studied. Now, if you want to address those questions, you need two requirements. So a model, an experimental model that has been subjected to the processes of domestication and breeding selection, and at the same time that is amenable for genetic investigation. Yes, obviously, sorry, I forgot to mention this point, because if we do know these genetics, we can rewire them for the benefit of mankind. Now, back into the requirements to study those processes, the model that we have identified in our lab has already been introduced by Simona, is barley, is a global crop, is also an excellent model for genetics and genomics investigation in cultivated plants for a number of reasons that you can read in this slide. And also, probably the most intriguing element of barley is that wild ancestors are accessible for experimentation, and some of them can be crossed, hybridized with modern cultivated varieties, and this led the opportunity to scientists to study those processes within the domestication and breeding selection framework. Now, we have a model. This is the way we study the interaction between plants and microbes at the root-soil interface. We apply a reductionistic approach. So we're looking at plants under controlled conditions. So we want to leave out what is the environmental variation to focus really on plant-soil interactions. And the vast majority of our investigations uses what scientists define cultivation-independent approaches. So we look at microbe via sequencing. And depending on the type of sequencing we perform, we may generate a census of the microbial population that are present in a particular environment, or we may try to reconstruct their functional potential and what they can do for plants. So by applying this approach in the early investigation, we were able to demonstrate that if you grow modern varieties and let's say wild ancestors side by side, what is a common garden experiment in the same soil, there is an effect on the microbiota that can be quantified, as you can see in these ordinations. So these are one of the most classical representations of the microbiota in many environments, and basically the key of interpretation is fairly simple. Every shapes represent a sample, and the position in the plot depict their, let's say, the relatedness of their microbiota. In other words, if you have shapes that are close to each other, they have a lot in common microbiota-wise. If you have shapes that are more separated, they have less in common. And by doing so, we were able to separate the three major categories that emerged during barley evolution. So the wild ancestor, the modern variety, and the landrace, that the evolution are somehow in the middle between them. And what we were also able to identify is that it's not just a wild versus domesticated scenario, but within, for example, the wild barleys, as a, let's say, sample, for example, in the fertile crescent, there is an impact on the microbiota. There are more than one wild microbiota, and there is more than one elite microbiota. And probably this has to do with the capacity of plants of adapting to given environment. Moving on, what we have identified is that this is not the capacity of plants of shaping the microbiota is not hardwired in their genome. So if you want, this is the ecosystem on a leash in barley flavor. meaning that if you subject modern and wild ancestor to fertilization with nitrogen, in presence of elevated nitrogen, their microbiotas becomes a lot more similar than what it is under, let's say, limited nitrogen conditions. So we went on and tried to identify whether this fluctuation in terms of microbiota recruitment represented by those bars reflected also some functional distinction of the microbiota, and this is what we were able to identify here. So it's not only microbes that are going up and down, but depending on the type of either modern variety or wild ancestor, there are different functionalities that are that the microbiota may exert and may provide plants with at the root soil interface. So we decided to apply what Bill told you a second ago, so a plant soil feedback experiment to try to understand what would be eventually the contribution of the wild microbiota to, for example, plant performance. The experiment is depicted here. So essentially, we conditioned the soil with either modern variety or wild ancestor, pull out the plants using the conditioned soil as substrate for the second cultivation, using an additional treatment. So we heat treated the soil that we have obtained in a sort of sterilization. So the soil is not sterile, has been sterilized, meaning that the endogenous microbiota that the two types of plants may have conditioned would have been disrupted in this scenario, or at least this was our hypothesis. So by doing so and using the modern variety as a recipient, we were able to demonstrate that, let's say, the endogenous microbiota, living microbiota, is much better than a microbiota that has been sterilized. And this is perhaps not a striking discovery, this has been reported by others. But ultimately, we were not able to identify differences between the two conditioning, so the wild microbiota and the elite one. And the reason for that, when we were looking at the microbiota in the second generation, was that essentially what is the current plants, so the plants that we have sown in the conditioned soil, was driving the recruitment of the microbes, regardless what would have been the let's say the input community. And this is something that in grasses has been demonstrated also in other species. For example, the work of Emilia Annula in the Netherlands is quite clear about this phenomenon. But this doesn't change the picture. So if the plants is driving the assembly of the microbiota, we won't be able to identify what would be the potential benefit of a wild microbiota associated with a modern one. So we decided to take a step back and try to identify what are those determinants of the microbiota in plants, because then in this case, we could combine maybe a modern plants with a wild microbiota in a more efficient way. And this is basically underpins the approach that we have followed. And these slides serve me also to inform you that for reminder of the presentation, I'll be using this color coding. Anytime you see blue or yellow, anytime you see blue is going to be the elite microbiota or elite barleys. Anytime you see yellow is the wild barley and the wild barley microbiota. So we have been using this, let's say, genetic population between wild and domesticated material and try to identify whether there was any segregation of the microbiota. And what you can see here is the result of these experiments. We could separate microbiota of the parental lines, and the population was somehow an intermediate level, more closer to what was the elite material. And this is due to the type of genetic population we have used, but there was no real clear, let's say, categorization. There was a more continuous variation. So we decided to apply quantitative genetic theory to try to see whether we were able to identify drivers of microbiota diversification. And this is the result of this investigation. So there are two pieces of information here. Here, what you're looking at are the 7 blocks. This is the barley genome. And we have superimposed here the classification of the microbiota. ESV level, if you're expert on this, this is basically the terminal node in taxonomic classification of the microbiota, while genus and family are, let's say, higher ranks. And what we have seen here, in a way, is the sort of ecological congruence that Simona was reported for the functionality at family level. So at different taxonomic level, there seems to be element of the barley genomes that condition the composition of the microbiota. The most striking one, at least for us, was located on chromosome 3H. And then for a reminder of this presentation, I'll be referred to this as locus 3H. Okay, so we have a map position. When you have a map position in genetics, you have only one way to go, that's by cloning. And to do so, we went back to the population, and we were able to identify a member of the population that had residual heterozygosity at the locus. meaning that if you self-propagate this plant, a portion of their progeny will have the locus at the, let's say, elite allele, and a portion of the locus at the wild allele, and the rest of it is somehow conserved. This is an ideal scenario to test this hypothesis. So if locus 3H has something to do with the microbiota, we should be able to see differences in terms of these sibling lines. And we were able to reject our null hypothesis. Indeed, we were able to separate the microbiota of these sibling lines, meaning that, at least in part, the locus is contributing to microbiota recruitment. This was for bacteria, but we couldn't see the same for fungi when we tested that, meaning that locus 3H, at least in the soil and the condition we tested, operates predominantly on the bacterial component of the microbiota. So a locus is a number of genes. So we reasoned that if we are looking at the, let's say, root expressed genes, basically what would have been the leftover of the microbiota profiling, this would have been the first port of call in terms of genetic recruitment for the plants. And we set up these experiments by looking at the genes differentially expressed in the roots of the sibling lines and our control line. We ended up with a relatively limited number of genes, and this is justified by the fact that we are operating with sibling lines. So the majority of their genome is conserved. So obviously, even their transcriptome will follow accordingly. But we know also where to look. I mean, besides these genes that are up and down, we know that the locus, the map position in terms of locus 3H may be the one to look for potential candidates. So obviously, we want genes that are mapping there. We have different lines that exhibit different microbiota recruitment. So genes that are differentially expressed and between them could be good candidates. And so are candidates, genes that are not differentially expressed between them, but they carry genetic variation within their sequences. We were able to identify a relatively limited number of candidates within fulfilling those criteria that we consider as primary candidates for the regulation of the microbiota and the barley rhizosphere. What we found particularly interesting is that if you look at the categories of these genes, some of them seems to match what are the functional requirements exerted by the microbiota in the rhizosphere of either wild or elite varieties. So one of these candidate codes for an NLR gene. If you are an expert in plant microbe interaction, you know perfectly what this acronym stands. For the people that are less familiar, let me bring back one, let's say for a moment, the way basically plants fend off potential pathogen using their innate immune system, in particular two categories of receptors, also called pattern recognition receptor, that Simona illustrates very elegantly in her presentation. Our candidate is actually intracellular one, and it's called NLR. And we wanted to know what happens if we were able to change this NLR at the microbiota thriving at the root soil interface. Nowadays, one of the most efficient way to do so is through genome editing. We were very lucky for two reasons. So the first one is that genotypes that are transformable, genetically transformable in barley, carry the same locus, the same 3H locus of our elite variety. So basically the NLR candidate. And when we attempted this for the first time, we identified immediately roughly 20 base pair deletion in some of our transformant at the 5' termini of the gene. So what happens is that this is a disruption of the potential functionality of the gene. So we have a, let's call it wild type allele and a disrupted allele. So this allows us to basically test this scenario, utilizing also as an additional control lines that were transformed, so subjected to the same process of our edited lines, but their genes was essentially a wild type one. And when we tested those plants in the same soil where we have grown our previous experiment, our previous mapping experiment, first of all, we identified that there were no obvious pleiotropic effect. Anytime there is a mutation in the plants, there is always the risk that alongside what you are interested in, there is something else that goes wrong, and then the plants, it becomes difficult to pinpoint which one is which. But our plants basically are indistinguishable between wild type. And when we were looking at the microbiota, so these growth promises are control, basically the wild type plant with the locus pH. This is the transformed lines that was not edited. This is transformed line harboring this deletion. And there is a clear shift in microbiota composition, with the caveat that these lines are still the Cas9 construct. Again, if you are into genetic transformation, you know what this means. And what we are doing right now, well, what we have done is actually prune out genetically this construct to have a better appreciation of what happens in terms of microbiota recruitment. So here we have a gene that is actually present in the modern varieties that once is disrupted, led to a reconfiguration of the microbiota. So if I go back to what I was telling you at the very beginning of my presentation, so the search of the potential wild Marley microbiota and wild genes that were missing from the modern variety, so this may suggest a different scenario, or at least a scenario that has to be considered. Second, why it doesn't move? Okay. Yes. Sorry, I had a moment of glitch. So alongside what has been lost to domestication, it is fair to consider there are post-domestication evolutionary innovation of plants. And in barley, this has been demonstrated with plant microbe interaction above ground that can indeed modify the adaptation to the environment. So perhaps, rather than looking only at wild genes that were lost, we need to reconsider modern genes and maybe disrupting them to unlock what is the potential of the wild barley microbiota. This is an area that we would like to investigate. Okay, this is the end of my talk. Basically, the main message that I would like to pass you is that the microbiota may be really considered, microbiota, the root soil interface, may be considered as an external plant phenotype, and of course can be studied and manipulated in the way other plant phenotype may be manipulated. So, open research questions. So, if we may have a culprit, so this immune receptor that shaped the microbiota, but we don't have the crime. So we don't know at the level of plant performance what happens having or not having a given configuration of the microbiota. And what Simona and Ben show you as a, let's say, the totality of the microbiota as a overall microhabitat, what you are interested in is basically looking at the handful of bacteria that are controlled by locus 3H, so the niche governed by locus 3H, and see what happens to the plants. And Katie, the first may have an answer for that. Then to be very specific, so I try to convince you that an intracellular receptor, like an LR, not only regulates something that is outside the cell, but is outside the root corpus. So ecologically, there is a link here that we haven't yet clarified. This is something we are working on. And the last one is something that becomes clear when I show you where we want to go. So if we were able to basically able to monitor microbiota and the interaction between plant and soil at the point of care, let's say in the field, I think there will be better positioning to try to deliver what are these anticipated impact in terms of sustainable production and sustainable development, as alluded also by the other speakers. With that, I'm closing. This is my team and the funding bodies that have supported this research. And the last one for you, very happy to take any question related to what I told you.
Right, thank you, David. So, applause. So we don't have a question in the chat for now. So what we can do maybe is anyone who wants to ask a question can raise his hands. Or again, I don't know if there's an audience back in Dundee. If there's people who ask questions, then maybe Ken, you can raise your hand as well. Right. So any question? Yes.
Simona has her hand up. In case you can't see that.
Okay, I can't see it. I don't think so. But yes, go for it.
Maybe I can start. David, a wonderful work. This is really beautiful genetics dissection. That's very, very good. So do I understand correctly that the allele that it is present in the elite has a higher level of expression compared to the wild allele? But and then you get a mutation in the golden promise. And so is the mutation in the golden promise mimicking the wild allele, or is the wild allele not active, or is it just the level of expression that has been modulated by the breeding? What do you think that?
There are multiple haplotypes. So the wild allele, so the parental line of our population has an expression level that is lower than the elite, but there are also loss of function within both the domesticated and wild germplasm. What our edited alleles try to mimic is this loss of function.
Mm-hmm.
Okay, so there are no questions in the chat, and one is related to, so I'm going to reframe it a little bit because these are long questions, but is there a possibility that these modifications that you observe to the plants and this locust that is modified, is there a possibility that there is an effect on root architecture and morphology, or maybe you can tell a bit more about what could you see in the phenotype that could have an impact?
Okay, we did not see a root architectural phenotype when looking at the root architecture of the sibling lines, so the lines that have the different alleles at the locus. I have to stress in the condition that we have tested, which implies the soil that we have tested. I can't exclude that maybe in different soils or in different conditions, the root architectural phenotype may play a role. Obviously, root architecture determines what kind of microbes and what kind of profile of soil plants engage with. So certainly this is a central point. But in this particular case, I tend to exclude our architectural phenotype.
So I have a question myself, which is when you present, so it's early on in your presentation, I think it's slide 7 or 8, when you present the difference between wild, landrace, and elite, and it doesn't look like there's a continuum. It looks like they're completely 3 different opposite conditions. And I'm wondering if, I mean, why is it not a continuum? And is it worth trying to understand what actually changed continuously to find out a bit more about, also from the microbiome perspective, what is changing mechanistically?
Okay. Yeah. So this type of ordination may be slightly misleading at times, okay, because obviously it focuses on the differences. So the genotype effect, even looking at, let's say, wild and say elite varieties, is in the order of 10–15%, sometimes even less. So the first principle is that the majority of the microbiota is conserved. Fraction of it is influenced by the genotype. So therefore, there is a sort of continuous, let's say, a continuous variation.
Thanks. So we have another question from Janice, who is asking whether the thing is changing. Whether you expect also an effect of this mutation on resistance to pathogens.
Possibly, this is one of the scenarios we are exploring. So I can't really name the pathogen right now because we don't know. So if, let's say, if you believe in what, let's say, in what I show you, in the sense that there is an element of selection operating on this NLR to domestication and breeding, so breeders never really selected for the microbiota. So this could be a trait that is conditioning others. Resistance to pathogen is one of those, yield is another. So possibly, as a consequence of selection for those, there is an impact in microbiota composition that we have yet to elucidate. So in short, yes, possibly yes.
Okay, so we have a question also from Tasmin Ahmed. So who is asking that in one of your slides you said no differential expression regarding one of your candidate gene, XTH. Could you please explain a little bit?
So essentially, when we run the RNA-seq experiments, we end up with a number of candidates or candidate genes within this area. So we wanted to define criteria to prioritize the one for further characterization. We had already in mind that we wanted to do a genome editing or let's say a mutagenesis of some of those candidates. But in the locus, there are right now 30 genes, and there were too many. So we need to reduce this number. So we thought that if a gene that maps there, even if the expression is the same between the sibling lines, but the coding sequence is different, so this may well be a good candidate for microbiota recruitment, because the proteins they code for will be different, and this may have an impact on the microbes.
Okay, thank you, Davide. So unfortunately, we have to stop now and to close this section. So we have the privilege to host Uta Pasovsky from the University of Cambridge. And Uta has made a really significant contribution to current knowledge on molecular mechanisms that governs symbiosis of Aberscola mycorrhiza. And today she will update us on integrating phosphate nutrient stages and Aberscola mycorrhiza. Right, Ute, down to you.
Thanks very much, and thank you very much for including me in on this special day, on this special event. Simona, full-hearted congratulations for you. Instead of flowers, here's a celebrations arbuscle for you. Well done, very well deserved. So yeah, I would like to give you an update on some of the insights we've gained in recent years about the integration of plant phosphate nutrition stages and avascular mycorrhizal symbiosis. So as not everybody might be familiar with the system that we work with, I included this slide here, which shows an ongoing avascular mycorrhizal symbiosis, so you can appreciate the extensive proliferation of the fungus surrounding the root here. All of these fine threads are fungal hyphae. The globular structures are fungal spores. And as much as an extensive proliferation we see here around the root, we need to imagine also occurs inside the root, leading to this very intimate association where the fungus intracellularly colonizes plant cortex cells, as shown on the previous arbuscle. So the outcome of the association is mutually beneficial, where the plant feeds the fungus with organic carbon, specifically with fatty acids, and the fungus at the same time enhances the phosphate nutrition of the plant. It's actually not restricted to phosphate. Also nitrogen, water, zinc, and other minerals are eventually delivered to the plant, but it is the effect is most pronounced for phosphate and can be explained by the dramatic increase of surface area for nutrient acquisition, for phosphate acquisition, that we see nicely illustrated here on this slide. So the benefit the plant derives from this interaction can be quite significant, as illustrated here under medium input field conditions. We're looking at 2 rows of maize plants that are isogenic and only differ in one gene being mutated here on the right-hand side in that row of plants. And that renders this genotype unable to engage with avascular mycorrhizal fungi in the soil. And you can macroscopically see that this comes with a penalty on crop performance, but it also comes with a penalty on yield. So we can basically, for the first time, put a number to the benefit plants derive from the ability to engage with these avascular mycorrhizal fungi. In this case, even under field conditions, when it comes to yield, we have a 30% yield increase in the wild type that is able to engage with these beneficial fungi. So you may wonder, I mean, there's an obvious potential here to make better use of this avascular mycorrhizal fungi for sustainable crop nutrition. So why is the symbiosis not more widely implemented in agricultural practices? And the reason for that is shown here, where we're looking at some extremely old data from the late 60s. And that already back then illustrated that there is an inverse correlation between the availability of phosphate to the plant and the degree of colonization. So basically, the plant monitors quite precisely what its nutritionist demand for phosphate is, and adjusts its competence or willingness, so to speak, to engage with the fungus accordingly. So one of our main research lines in the lab is trying to understand the molecular mechanisms that underpin this competence of the plant to engage in this association, and in particular, of course, with regard to how this phosphate nutritional status is integrated in that competence to establish an avascular mycorrhizal symbiosis. And for many years, we've been working on this receptor protein, which is an alpha beta hydrolase in rice, called very uninspirational dwarf 14-like. This is, for reference, this is the evolutionarily older paralog of the strigolactone receptor. It's an extremely old gene, so you would, or ancient gene, you will find a prototype actually in the green algae and the ancestors of the land plants. So we first had a mutant and then eventually cloned the gene from this mutant. And when you don't have this alpha beta hydrolase, you can see here on the micrograph that basically the rice plant is completely unable to engage in the association with avascular mycorrhizal fungus, in this case Rhizophagus irregularis, as opposed here to the wild type, where we see different fungal structures, hyphae, vesicles, arbuscles inside the wild type root. No such thing can be observed in the case of the mutant, and such a phenotype could be due either to the fact that the mutant is no longer able to perceive the fungus or is unable to prime the fungus, to activate the fungus. And doing RNA-seq on mutant and wild type roots to monitor the transcriptional responses to fungal exudates, when the roots were treated with fungal exudates, we found that there is indeed a diagnostic transcriptional response in the wild type that is missing from the mutant. And from that, we concluded that the dwarf 14-like gene is required for the susceptibility of rice to AM fungi. Now, when and where do we require this receptor to be active? So Gabriel Ferreras is a freshly baked postdoc in the lab. He did his PhD with us and just defended yesterday successfully. Congrats, Gabriel. And he made use of a set of different promoters that drove the expression domain into different root tissues. So we have the native D14-like promoter, which is a bit heterogeneous and covers anything from steel to eventually outer root cell layers, but in a rather stochastic pattern. Then we have a dedicated steel promoter and also a promoter that encodes a phosphate transporter that where promoter activity is predominantly active in the outer root cell layers. And so he used these promoters to drive the D14-like gene and try to complement the D14-like mutant, and found that, as expected, the native promoter was able to bring colonization of the mutant back. So we have normal classical complementation here. We're looking at different fungal structures inside the root as one of the typical ways to quantify fungal colonization of the root. We see that the steel promoter fails to bring full complementation back. We have partial complementation relative to the mutant. But then, quite convincingly, we see that the phosphate transporter promoter driving expression in the outer root cell layer fully complemented the mutant phenotype, and therefore we conclude that D14-like is required in the outer root cell layers, which is consistent with a role in facilitating root engagement with avascular mycorrhizal fungi. So a former postdoc in the lab, now group leader at the Crop Science Center, Jong-Min Choi, she picked up on what was known about this receptor from the Arabidopsis world. So the D14-like receptor in Arabidopsis is called Chi2 for Carrikin insensitive 2. And in Arabidopsis, the receptor had been well characterized in the context of development, but what we actually learned from the colleagues was that upon activation of the receptor, a receptor complex would be recruited consisting of an F-box protein, which in rice is called D3, and a negative regulator, SMAX1, that becomes ubiquitinated and sent to the proteasome machinery for degradation. So basically, we need to get rid of the negative regulator, SMAX1, to de-repress the developmental programs. And Jong-Min was then able to show that the same kind of signaling mechanism also is operational in symbiosis in rice. Here we see the D3 mutant phenotype. We see a dramatic reduction of colonization relative to the wild type. And when we now turn to the SMUX1 mutant, we see the opposite. We see a significant enhancement of colonization relative to the wild type, which is consistent with SMUX1 being a negative regulator of colonization in rice. And then to address epistatic relationships, Zhongmin also produced a double mutant between D3 and SMUX1. And you can see that introducing the SMUX1 mutant allele into the D3 mutant background suppressed the D3 phenotype. So this together told us that A, SMUX1 is a negative regulator of symbiosis, and B, that it functions downstream of D3. So this is really a type of signaling mechanism that we're quite familiar with in plants from different hormonal signaling pathways. We basically have a double negative regulation, and what this further led us to hypothesize was that most likely the activation of the D14-like receptor is therefore needed to get rid of SMUX1, hence the mutant phenotype that we saw in D14-like and in D3 might be attributable to a stabilization of SMUX1, and that was indeed the case. Here we're looking at the Western blot that Zhongmin did, and you see that the SMUX1 protein is indeed stabilized in these mutant backgrounds. So what I would like you to also notice is that we at the same time do not see any detectable levels of SMUX1 in the wild type, which seems to indicate that under wild type conditions, this pathway is on, leading to the removal of SMUX1. And that really changes our way of how we're looking at these initial signaling events in the symbiosis, because we keep thinking about the need for an activation, and what actually these data would rather point to is that, at least with regard to the central signaling pathway, we have a competence for symbiotic engagement continuously on, but there is a pathway, this pathway, that has evolved to potentially put a brake on, perhaps under unfavorable conditions. So Zhongmin went ahead and also wanted to know what is it that SMUX1 is eventually blocking. So she did an RNA-seq experiment and then found a strigolactone signature in the transcriptional response, the upregulated gene list in the SMUX1 mutant, and then found that basically the entire biosynthetic pathway leading to strigolactone was induced when SMUX was mutated. And we collaborated eventually with Haro Baumeister to also address whether or not that transcriptional response is actually translated physiologically in an increased production of strigolactone here for deoxyorobanco. And just to draw your attention to the D3 mutant here, you see that is the amount of strigolactone detected in D3, and then in the double mutant we indeed see that massive increase when SMUX1 is mutated. So that pointed to a crosstalk between the D14-like signaling pathway and strigolactone biosynthesis, which at the time was new. Zhongmin furthermore realized that there is a symbiosis signaling signature, namely a number of genes that we know are components of a symbiosis signaling pathway are transcriptionally induced. And just as a very, very rough sort of explanation of the symbiosis signaling pathway, what we would be looking at is receptors in the plasma membrane perceiving the fungus that leads to the production of a secondary messenger and the induction of nuclear calcium spiking, which is deciphered by a calcium and calmodulin dependent protein kinase that then drives the genetic program further for symbiont accommodation in the root. So we were asking the question, Rafaela Hull, a former PhD student in the lab, is the D14-like signaling pathway indeed upstream of the symbiosis signaling pathway, as was indicated by that transcriptional induction of a set of these genes displayed here on this cartoon? And what Rafi did, she produced an autoactive version of this deciphering calcium and calmodulin protein kinase and introduce that gain of function construct into the D14-like mutant to address epistasis, and also into the kinase mutant itself. You see the construct is functional. It brings colonization back in the mutant, and it does so as well in the D14-like mutant, pointing towards the D14-like signaling pathway functioning upstream of the symbiosis signaling pathway. So that brings me to a sort of intermediate summary. So what we believe is happening, this is our model, is that under favorable conditions, there is a yet unknown ligand of the D14-like receptor that leads to the activation of the receptor and the recruitment of the receptor complex, which in turn leads to the degradation of the SMAX1 protein that liberates critical transcription factors that now drive the strigolactone biosynthesis and also common symbiosis signaling pathway genes. So combining the attraction of the fungus through strigolactone production and secretion and the activation of the accommodation program. And that leads in the wild type to a competence to engage with the fungi. When we then genetically completely remove SMAX1, we actually do see this over colonization of the root. However, when D14-like is not activated or is absent, or the F-box protein D3, SMAX1 is stabilized and blocks these critical transcription factors, and we indeed, in both cases, don't see interaction. And now the question for us was, is actually high phosphate fertilization, is that one of those unfavorable conditions that would block symbiosis development? And that brings Thomas into the game. He is a postdoc in the lab, and he was then interested, in order to address that question, to actually follow SMAX1 degradation or stabilization. And he produced a ratiometric SMAX1 degron approach, whereby he used a strong constitutive promoter to drive the key domain that is required for interaction with the receptor, translationally fused to a red fluorescent protein, followed by a ribosome skipping sequence and another fluorescent protein, in this case YFP NLS tagged. And what would happen here is that the ribosome would read that SMAX1 RFP part of the RNA, then drop off and come on again and produce a second protein at equal ratio to the first, and thereby you create a ratiometric degron strategy. So Thomas also included a deletion variant of that domain, which is no longer able to interact with the receptor and therefore should be constitutively or constantly stabilized. And he introduced these constructs into protoplasts to verify that they're functional, and that is indeed the case. As you can see here, we have the wild type domain. We see the YFP in the nucleus, and we don't see actually SMAX1 wild type domain consistent with being degraded in this scenario. However, when we have the deletion variant, we now do see stabilization of the SMAX1 domain in equal ratios to the YFP. So Thomas then transformed these constructs into plants. So we have stable rice plants that now allow us to actually monitor SMAX1's protein stabilization across the plant tissue. And you can appreciate that we have a gradient here between a higher stabilization of SMAX1 in the root tip, and then slowly but surely less stabilization higher up. We also have a proximal distant gradient here across the root diameter. So this gives us now a really, really high level or high resolution level of monitoring SMAX stability. And you start to see really interesting patterns, as shown here on the left-hand side, where we see that lateral roots actually show a higher level of degradation of SMAX1 than the crown roots. So now Thomas wanted to know, is SMAX1 stabilized in response to elevated phosphate concentrations? And the answer is yes. We're looking here at low phosphate, high phosphate of the wild type and the deletion variant, and across the 3 different root types of rice. And you can immediately see that for each of the root types, there is indeed an increase in stabilized SMAX1 protein when more phosphate is available. So that now prompted us to think along the line of what would be the effect of SMAX1 mutation on actually the suppression, high phosphate suppression of colonization. So if SMAX1 is stabilized under high phosphate, it may explain that suppressive colonization that we see. If we take SMAX1 out of the picture, do we get more colonization? And that was spearheaded first by John Min, and then Thomas followed on the heels. And here we see the data for how the colonization is eventually suppressed from low phosphate to high phosphate in the wild type. And indeed, when we mutate SMAX1, we see that colonization is still suppressed to some extent, but way more colonization is enabled relative to the wild type. In the meantime, we have introduced the SMAX1 CRISPR construct also in other rice varieties and indica varieties, and we can confirm that same phenomenon is coming back repeatedly, consistently. So this is one of our engineering targets, to ensure continuous colonization under variable phosphate fertilization regimes. So in summary, I think we are quite confident that as SMAX1 stabilization occurs in under increased phosphate fertilization conditions, that we indeed have an explanation for the unfavorable condition here, matching the high phosphate condition. And what I would like to really emphasize as a last comment here is that it is an incredibly elegant way nature has invented to integrate phosphate nutrient status of the plant with, at the same time, then inducing the attraction of the fungus through an increased production and secretion of strigolactone, and also at the same time opening the doors to accommodate the beneficial fungus. So this is the gang at the moment. I would like to specifically highlight here our fantastic collaboration with Emma Wallington, who does all of the rice transformations. Zander Jones, very, very helpful in the design of the Degon construct, Haro Baumeister strigolactone analysis. And thank you for your attention. I'm happy to take questions. And congratulations again, Simona.
Thanks very much, Uta. I mean, lots of work here that you've presented. So we're now moving to questions. So I'm going to look at the chat. So Tim George is asking, does the high level of SMAX stabilization in the root tip suggest that the plant is trying to avoid AMF colonization of that new tissue?
So yes. Oh, now there's an echo. Is it okay? Yeah, no, it's okay. All right. Yeah, so basically that is what we would find, that when you have high phosphate fertilization, new tissue is very limited or not colonized. Yeah. Now you are muted. Now you're muted.
Yes, I didn't unmute myself. So Simona, do you want to ask yourself directly, maybe?
Yeah, wonderful work, Uta. This is such a fine genetic dissection. So I don't recall, maybe I should look back at your work. Is there a specificity between the D14-like receptor and different AM fungi? So is there, in other words, are there other AM fungi that can bypass or have different requirements for the presence of D14, or is it is a real entry for any kind of AM fungi?
Yeah, so we routinely use phylogenetically distant fungus as our main comparative fungus, as a member of the Gigasporoaceae. So back when Caroline did the first characterization of the mutant, we use Gigaspora rosea, and there's no difference. So, and from the majority of genetic material that we work with, we don't necessarily see any major difference, significant difference between colonization, extent of colonization, and between the different type of fungi. Yeah. So it should be very, very broadly, applicable to mycorrhizal fungi, what we observe. Yeah.
So I had a kind of similar question about how broad this was, and maybe I'll take it from a different angle. When these field trials and this maze that you're showing, or the others field trial that you might have run, do you attribute the difference in growth and physiology solely to the presence of the MA fungi, or and phosphorates, or are there other factors that might be there?
Yeah, thank you both for these questions. So there is, if you let's say take one fungus and a set of genetically diverse rise lines. So we've done that. Emily Savante in the lab has done that. You see a huge difference in the outcome of the plant performance as a result of being inoculated with the fungus. Anything from growth depression to massive enhancement of biomass in response to colonization. So that means that we are expecting that there are genetic factors that we don't know yet that define the extent of of benefit, or respectively the outcome of the symbiosis with regard to crop performance. What I should emphasize is that this is not correlating with colonization. So colonization variability does not correlate with variability in this benefit. Of course, having said that, you need to have a certain threshold, a certain minimal amount of colonization to see a benefit. That's clear. No? But then if you drive it through the roof, then you don't necessarily see a higher benefit. So what we hope to achieve with this particular engineering approach is just to make the colonization less vulnerable to the regulatory mechanisms that the plant has evolved to control colonization relative to the nutrient status, so that we have colonization.
Okay, so I think we're getting to the end of this session, and I see Ken turning up on the, indicating me, please finish. So Ken, I leave it on to you now, I guess.
Yeah, apologies. I was trying to be subtle there, Nell, but I think you called me out on that. So thank you very much. So, yep, we'll break now for lunch. But yeah, just to say thank you very much to all the speakers from this morning. I think all fantastic talks, and thank you everyone for engaging and asking questions after each of the talks. So we're now at the point where we're going to break for lunch. We'll reconvene at 2 o'clock UK time, so round about 54 minutes from now, and then Tracy Valentine will be chairing the afternoon session, where we'll start with 5 flash talks pre-recorded, and then there'll be an opportunity to ask the speakers or people who delivered their flash talks some questions before we have open the afternoon's full talks with Corné Peters, Plant Microbiome, the Extended Host Immune System. So, yep, a quick break for lunch, and then see you all at 2 o'clock UK time. But thanks again to all speakers this morning.
From Maria Hennis Soriana.
Carmen Esquiado Martinez, Daniel Patco, Max Danson, and Katie Arnton. So, Ken, would you like to do your stuff?
I will try. So, find it. So I will say, if just shout, Tracy, if you don't hear the sound from the video.
I'm Maria Hernandez. I'm a researcher at Dundee Center, and I'm providing a snapshot of a field trial that was run to link the WIS routes and Route to Rest project's activities. And it was a collaboration between myself at Dundee Center and Tim George team at the James Hutton. So the idea was to screen 10 bred wheat genotypes, including 8 land races with potential to decrease nitrification inhibition, and 2 modern cultivars used as a control, plus 10 different barley genotypes, also selected for different potential to control nitrogen cycling in soil. So these 20 different genotypes were tested in the field in triplicate for 2 years consecutive, using 2 different nitrogen fertilizer regimes. And then we collected both bare soil and rhizosphere soil from the different plots, and we performed analysis for 16S and ITS. Plus, during the first year, we also measured potential nitrification rates in the same soil. We only found marginal differences between the genotypes, while for the nitrogen content in seed, particularly for the barley genotypes, we found quite drastic differences between the genotypes when compared to the modern cultivar SASI. Looking at the impact of the different genotypes and the different conditions in the rhizosphere microbial recruitment, we can observe in this graph that both, when we divide the genotypes between barley and bread, we observe a very significant difference in the recruitment of the microbiome in the rhizosphere, and a lot of this diversity is also quite driven by the nitrogen dose applied and only marginally by the cultivars, by the genotypes. If we look separately just at barley, we can see that there is a quite a strong difference between the 2 nitrogen doses applied in the field, and much less influence of the actual genotype, while for the bread cultivars, we observe less impact of the nitrogen dose in the microbiome recruitment, and a little bit more of impact due to the genotype. Thank you.
Ken, are you managing to upload the next one? We seem to have, okay.
Hello to everyone. I'm going to talk today about integrative crop trait profiling enables proxy-based prediction of host microbiota interaction in Vicia fava. The rationale is that rhizosphere traits remain underused in breeding because the measurement is technically difficult. Often they are low throughput and destructive methods. Here we develop a novel sampling approach for simultaneously assessment of rhizosphere microbiota and root exudates, and this approach can be summarized here and can be found following this QR code in bioRxiv. We use Vicia fava as a legume model to benchmark this new sampling approach and answer the question, can rhizosphere and root traits predict shift in the rhizosphere microbiota? So using this novel method, we can assess different classes of rhizosphere-associated traits, such as root morphology, root exudation, rhizosphere size, biomass, and nodulation. We found that high-throughput root exudation traits negatively correlate with plant biomass, but also nodulation. Then we use these rhizosphere-related traits as explanatory variables and the microbiome composition across soils as response variables in this redundancy analysis. Arrows will represent traits and dots represent the microbiota composition across soils, and different colors are the different soils. So we can see that the model is significant, and only these rhizosphere-related traits, they account up to 31% of the variation. And, for example, if we observe the soil bullion field, here represented in green, we observe that root spatial traits, they seem to be at least in part responsible for the composition on this microbiota in this soil. So with that, I would like to thank to everyone involved in this work package of the Horizon Europe project Root to Rest, and you for your attention. Thank you.
Dear audience, in my presentation I'm going to show you two of our latest developments to observe and model rhizosphere processes. In our lab, we work to develop new technologies to quantify the rhizosphere processes, such as the organized and collective movements of soil bacteria. Recently, we decided to work with microfluidic technologies because microfluidics offers high throughput and highly controlled measurements. The first example is a filter paper-based microfluidic system. Filter paper is a very simple material, and with a very small modification using wax printing, it is possible to create hydrophobic barriers in it, creating parallel fluidic channels. The root can grow over these channels, releasing exudates into the paper structure, and using capillary flow, it is possible to extract the exudates and deliver them towards sensors, infield colorimetric and calibrated sensors, or it is also possible to cut off these little paper parts for further analysis using HPLC, for instance. Based on this very simple technology, we could reveal the exudation differences along the root over time. We could show the exudation differences between wheat varieties. However, these wheat varieties looked exactly the same. The other technology is also from the microfluidics domain. It's a photopolymer-based channel system. One channel has a nutrient source, the other channel has the bacteria. And through this photopolymer membrane, the nutrient can diffuse to the bacteria channel, functioning as an exuding root. And based on the concentration of the bacteria and based on the nutrient source, we could observe very different bacteria behaviors in the system. So in my presentation, I wanted to show you these two technologies. If you have any question, please don't hesitate to ask. Thank you for your attention.
Good morning, I'm Max Tasse and I'm here to talk about a root-secreted coumarin that improves iron nutrition of a plant beneficial Pseudomonas. So, coumarins are these fluorescent compounds that dicotyledonous plants secrete when they are experiencing iron deficiency, and they're produced by F6H1, which leads to scopoletin production, which is then converted by SH into fraxitin. And when they are in the rhizosphere, they change the composition of the rhizosphere microbiome. And since they are antimicrobial, certain microbes are more resistant to them. In this case, WCS417, our lab's pet strain, plant beneficial Pseudomonas, than other microbes. So in this case, Fusarium oxysporum, a soil-borne pathogen, is more susceptible to these coumarins. So we wanted to investigate what determines this WCS417 coumarin resistance. And we test this with scopoletin and fraxitin, the main secreted coumarins. So we tested a WCS mutant library and subjected it to coumarin pressure to identify which mutants don't grow well anymore in the presence of coumarins. And this way we identified 41 fraxitin tolerance associated genes, while for scopoletin we didn't find many. So we focused mainly on this fraxitin set, and we identified that the cell wall, function unknown, and inorganic iron transport and metabolism had the most number of genes associated with them. And we were especially interested in the last one because we found 2 TOMB-dependent receptors, and we know that these fraxtin can benefit plants in their iron nutrition and that they can form chelates with iron. And we thought maybe fraxtin sequesters part of the iron pool that then becomes unavailable if you don't have these iron-related genes, such as TOMB-dependent receptors. So we went to a minimal medium setup to see if 417 can utilize iron in the presence of fraxtin, and we see that at the exponential growth phase, the fraxtin can actually improve the growth of the wild type, and it can rescue the lagging growth phenotype of the pyoverdin mutant, which lacks its main cytosine. We don't see this when we don't add iron. And if you take the pyoverdin mutant and subject it to bipyridine, which is a strong iron chelator, it can't grow, but it gets rescued in a dose-dependent way by fraxtin. So in conclusion, 417 iron nutrition genes play a role in fraxtin tolerance, and fraxtin can aid 417 in iron nutrition. And the next steps, we want to test these TOMB-dependent receptor mutants. With this, I would like to thank you all, and that's it.
Hello, I'm Katie, a PhD student in Davide Bulgarelli's lab. The rhizosphere microbiota, the community of microbes found in the thin layer of soil surrounding the roots, is not randomly assembled. Plant genes control a first differentiation from unplanted soil, known as the plant effect, which is then fine-tuned at individual genotype level, creating the genotype effect. I have been working on a protocol to study these interactions in the model crop barley by using synthetic communities, or syncoms for short. Syncoms are collections of co-cultured microbes that act as a simplified mimic of the natural microbiota. I sterilize and germinate barley seeds and create a syncom inoculum by combining 8 bacteria previously isolated from the roots of barley. I combine the seedlings and the inoculum in sterile growth tubes, creating a notobiotic environment where every organism interacting with my host plant is known. After 2 weeks, I harvest my plants, collecting the roots and shoots to measure their dry weight and a sample of the rhizosphere to generate a 16S rRNA gene sequencing library. The 16S rRNA gene is a phylogenetic marker for bacteria, letting us know what bacteria is present and their relative abundance. I'll show you some of this data now. In this cat plot here, each point represents the bacterial community of one sequence sample. Points closer together have more similar communities, and points further apart have more distinct communities. You can see 3 distinct clusters. These points in grey represent samples of the inoculum. These colourful points represent rhizosphere samples from plants inoculated with the syncom. And these black points represent samples of unplanted sand, which have also been inoculated with the syncom. The distinction between the planted and unplanted samples is our plant effect, which accounts for over 90% of the variance between our samples. If we look closer at just our planted samples, we can see a more limited genotype effect, accounting for around 17% of the variance between the planted samples, mirroring what we would expect to see in the soil, but with a far simpler community and under notobiotic conditions in the lab. You can see the community of the genotype in pink is a bit different than the communities of the other 3 genotypes. If you would like to find out more, including the effect on plant growth, the drivers behind the plant and genotype effects, or how the balance of the inoculum affects colonization, I am currently writing up this research with a hope to publish at the start of next year. Thank you.
Okay, so that's our 5 flash talks. We've got at least one question in the chat, and I've got another one. But please put your hand up if you've got other questions. So we start with one from Tasnim. Are you able to just unmute and ask your question? If not, I'll ask it. So the question was aimed at Daniel, and it was asking how you collect the everything's moving. How you collect the exudates from the filter paper.
Thank you very much for the question. It's a very simple method. I take the chip and put it horizontally into a petri dish. I add little water into the petri dish, and evaporation on the other side of the paper channels will generate a continuous slow flow in the paper structure. And that capillary flow, evaporation-driven flow can deliver the sample into the inbuilt colorimetric sensors, or it is also possible to cut off this little outlying paper part and dissolve the sample from it for HPLC. I hope it was clear. I can share my screen to provide a better explanation.
Yes,
if it is necessary.
sorry. Yes, can you please repeat it again?
Yes, okay, if I am allowed, I am sharing.
Yeah, please do for 1 minute, Daniel.
Entire screen. Yes. So, yep. I'm sorry. So here is the chamber itself. So when after 6 days of incubation, I take this chip and put it into a petri dish vertically like this. And I insert
your slides.
You cannot see.
If you could just try and explain it verbally, then.
Yes, okay, I'm trying. So there is this chip with the parallel paper channels divided by the wax parts, and when I want to extract the exudates, I just need to put this chip into a petri dish vertically, horizontally, so each of the little paper channels can be contact with water, flow into the paper, and on the other side of the channel, the evaporation generates a flow inside the paper structure, and this flow can deliver the samples to the inbuilt sensors. This is one way, or I can simply cut off these little outlying paper parts and dissolve the collected samples from it.
Okay, thank you, Daniel. We've got a question for Katie from Tim George. How do you explain the change of community from the inoculant to that seen in the sand control?
Yeah, hi. So in my inoculum, I freeze it immediately after inoculation, whereas the sand controls have had 2 weeks in the tube to interact with each other and grow. So essentially, my inoculum samples are the balance that I assemble my inoculum in, whereas the sand samples are the result of competition and interaction between my bacteria without without plant present over time.
And is just a question follow on from that, is the sand sterilized before you use it, or would it have
yes.
a slightly natural population anyway? Okay, yeah, thanks. And so thanks very much, Katie. So we've got a next question is for Max. Is this a specific type of TON-B gene? What is its prevalence in other bacteria stroke taxa? Got Max?
Is it working?
Yeah, we can hear you, Max. Yeah.
Okay, great. Yes, so it's actually 2 TON-B dependent receptors we pick up. And we have also done a genetic association study in multiple pseudomonas, and at least amongst these pseudomonas, one seems to be more conserved than the other. So we haven't looked farther outside of pseudomonas yet, but this is part of the plan to do. Yeah. Thank you.
Thank you. And I'll go back to Katie. How different are the host genotypes from each other?
So the 4 host genotypes I've been using, some of them will be mentioned by Davide in his talk earlier. So we have Barker and Elite line. We have 2 sibling lines that Davide talked about. These are the ones with the introcretions and the wild lines at the 3H locus. And then we have ModX. which is an elite line that's more distinct from Barker and the sibling lines. So Barker and the sibling lines are quite genetically similar with the exception of that section of the H. And then the MODX is a little more different. And it's the one that we see having a more distinct community from the others.
Okay, thank you. And then I've got, going back to Daniel, are any of these techniques suitable for studying active metabolites in plant roots during pathogen infection, like nematodes, under soil-free conditions, so that the metabolite changes can be observed more clearly? If you could share your thoughts on this.
I really hope so, because I am planning to do it.
Anything else? Okay, so so I also had one, if I get the name right, I think it was Max. So so there was a screen done, and you've got 41 for the Fraxilin and 4 for the Scopoletin that you identified.
Yes.
Did you have any hypothesis?
That's correct.
Why there was such a big difference in the numbers that you?
Yeah, so what we also, and I will refer back to the genetic association study with the Pseudomonas that I was talking about in the first question. So we also tested in that screen multiple Pseudomonas for scopoletin resistance and Fraxilin resistance, and it seems that amongst Pseudomonas it is relatively conserved. So we also tested other rhizosphere isolates, and there it seems more spread out. So there is something in Pseudomonas, so it seems a quite robust, maybe redundant resistance that is difficult to detect with single knockouts, I assume.
Great, thanks very much. So that's all the questions that have been put in the chat. Is there anybody online that would like to ask a question? If you can put your hand up at all. No. Okay, then that's great. So thank you very much for all those people preparing their flash talks in advance as well. And we'll move on to Corne for the next talk. So if you could share your slides.
Yes, I can try to do that. Let me see. That's the wrong one. Cancel. Optimize. What do you want to share? Okay. Share. Window. This one, I think. Do you see it?
Yes. So
Yeah.
this is Corne Pietes, and it's going to talk about plant microbes, microbiome, and the extended host immune system. So off you
Yes.
go.
Thank you very much, and you know, I'm really happy that I'm part of this nice collection of talks in let's say acknowledgement of Simone, which is of course highly deserved that she got this nice award. So I'm a little bit in the same field as Simone. I'm working in the plant microbiome, and we basically look at the microbiome from the perspective that it is part of the extended plant immune system, and I'm going to talk a little bit about that. And what you see here is the group that we have. 2 weeks ago we had a retreat. As you can see, it worked out nicely. Everybody is still having fun together. But you know, if we think about microbiota, that is of course what we are going to talk about today. We all know that, you know, plants become covered with microbes. They can come from the seeds, but when the seed germinates, it picks it up from the soil. But it can also come from the air with insects, and at a certain point in life, the plant assembles different microbiota on different parts of its plant body. And if you look very carefully, and Simona mentioned that already in her talk, that when plants are, let's say, longer interacting with microbes, they influence which ones that they like to keep and which ones that they would like to deter. And if you look in the rhizosphere, you will see that especially exudates are very strongly able to shape the microbiome and well, determine in that way who is allowed to grow in the rhizosphere and which one not. And the same also basically also for the phytosphere. And for instance, if you look at the potato, and I'm going to talk a lot about potato today, let's say you take a potato and you look at the eye or the sprout or the peel or the flesh, and you look at the microbiome of all those different components, you will see that each component of a plant has different microbiome composition. So basically the plant really shapes the microbiome, well, often to its advantage. Because many of these microbiota have an effect on the immune system. For instance, some of them can produce antibiotics and inhibit growth of pathogenic fungi in the soil, for instance. Several have also been demonstrated to stimulate the immune system, leading to induced systemic resistance, and many examples of how beneficial microbes can contribute to the defense of a plant. And that's why we call that the extended plant immune system, as it adds up to the genetic resistance that the plant already has. And in 2026, there will be a nice overview coming out for on this topic. But you know, that's not all. It's not only the immune system. Many microbes, of course, also help out with nutrient uptake. Simona talked already a lot about the nitrogen, but also enhance stress tolerance, growth promotion. We can really benefit a lot from the plant microbiome. And in the past, we talked a lot about well, how single microbes can do that. And I show one example of our model strain that Max already talked about, Pseudomonas simiae WCS417. It's a very good colonizer of plant roots. When it does so, you see that the root architecture changes, many more lateral roots. Root hairs elongate, which is an auxin effect. If you use an auxin reporter, you see a stronger auxin response in the roots. It leads to enhanced resistance against, for instance, Fusarium oxysporum. Here you see a susceptible plant. And if you colonize the roots with the Pseudomonas bacteria, it becomes more protected against this pathogen. And Max already talked about the fact that the plant roots start to produce coumarins and secrete coumarins. They have an antimicrobial effect. The Pseudomonas itself is completely tolerant, but others are sensitive. And that is also one of the ways how the plant is able to shape the microbiome around its root system. But I'm not going to talk about that today, because I would like to zoom in a little bit on the power of plant phenotyping for future plant microbiome research. We have done a lot of phenotyping lately, and we also try to use that information to do microbiome-informed prediction modeling. So can microbiome profiles say something about the phenotype of a plant, and how can we use that in well, maybe in plant breeding in the future? So I will give a couple of examples of that. And well, we have a very nice phenotyping center in Utrecht. It's called NPEC. I will talk about it more often. Here you see a nice phenotyping example of potatoes growing in that phenotyping center. They're dancing over the day. If you look at the way the leaves are, let's say, behaving early in the morning and late in the evening. Okay, but let's zoom a little bit into the plant topic that I would like to zoom into today, and that is potato. We do a lot of work on potato, and the Netherlands is a very important potato country, like the UK as well, and Denmark actually too. But if you look at it, 25% of the arable land in the Netherlands is used to produce potatoes, and basically seed potatoes, because seed potatoes are the potatoes that are put in the soil to produce consumers' potatoes. It's a clonal way of reproduction, and what appears is that the location where the seed potatoes are produced is apparently very important for the potato vigor in the next season. And that's a very important topic for potato breeders such as Averus Seeds and HZPC. And what they observe is when you put a potato genotype in one soil or in the other soil, and you harvest the seed potatoes, and you then would put them in the soil the next year to produce more potatoes, then it depends on where the seed potatoes were produced, whether you have a high vigor or a low vigor of your potato. And they wanted to know, is there a effect of the microbiome of the soil in which these seed potatoes were produced? Nobody knows. And they said, well, can you sort that out for us? And we said, yes, if you show us the money, we will give you the answer. And so they showed us the money, and we showed them the answer. And that was published earlier this year in Nature Microbiology. So I will go through it quickly, only the essence. And if you want to know the details, I would suggest that you visit this publication. But basically what we did, we took 360 seed lots, so that's basically 360 fields where seed potatoes were produced from 6 different varieties. And what we did, we harvested all those seed potatoes. Of course, each soil has a different microbiome, so you can expect that these potatoes have different microbiome compositions from the seed lot where they were produced. And what we did, we put them in 3 different trial fields in the next year, 2 in the north of the Netherlands, and 1 in the south of France. And what we did, we monitored the outgrowth of the seed potato of all those different seed potato batches. And then you can say something about the vigor, because the faster the outgrowth of the potato, the higher the vigor. And it depends on from which seed lot it was produced, whether the vigor was high or low. And here you have a nice example. This is variety F, this is a certain variety of potato. And here you see seed lot number 249, that is a particular seed lot from which the seed potatoes were produced. If you put them in these 2 northern trial fields or in the southern trial field, they always produce higher than average. And if you take from seed lot 259, they always produce lower than average. So apparently this seed lot is imprinted to have a low vigor, and this seed lot is imprinted to have a high vigor, even though the genetics are exactly the same and everything in between. So what we then did is, of course, analyze the microbiome profile on all those different seed potatoes. And then, of course, you can say a couple of things already that are logical. Plant genotype matters. These are 6 different varieties, and you can already see that the microbiome composition on each variety is different. So apparently, the plant genotype shapes the microbiome composition on those seed potatoes. And also the environment matters, you know, whether it comes from the sandy soil or from a clayey soil, you also have a different microbiome composition. But that was not the intention of our study. We wanted to know the microbiome profile on all those different seed potatoes. To what extent does that relate to the outgrowth of the seed potato in the next season? So is there a correlation between microbiome composition and potato vigor? And Yang, the investigator on this project, together with Roland and with Juanjo, they made a prediction model based on the information that came out of this research. So we had all the drone images telling something about the potato vigor and the microbiome information. We put it all in a model, and there's many more figures in the publication, but the bottom line is, if you have the observed potato vigor and you have the predicted potato vigor based on the microbiome composition, you can nicely predict what is the vigor of your seed potato based on the microbial composition on that potato tuber. So basically what we conclude from this, the microbiome profile on a potato tuber can predict its vigor in the next season. That's of course nice to know, but what you can also extract from this information, if you have this model, you can also extract from the model which microbes contribute most to that prediction. So which microbes are most correlated with the fact that the potato is highly vigorous or lowly vigorous. That's something that comes out of this figure. And what David is currently doing, he is isolating the microbes that are mostly correlated with the potato vigor, and we're using a machine for that called the prospector. And what the prospector basically does, you take a sample, you put it in a machine, and the machine makes single isolates from that sample, and then you have, let's say, within 1 or 2 weeks, you can have a couple of hundreds of different isolates to make a biobank out of that sample. And what he, of course, will do is test whether, if you apply those microbes to the potato tubers, whether they increase the vigor of the potato. Well, knowing that this prediction modeling works, Young thought, well, can we try it also by making use of synthetic communities? We heard, of course, Simona also talking about synthetic communities. So what Young asked, okay, if we start with a collection of 50 fully sequenced bacterial strains, we know exactly all the genes, and we make different combinations of 9 out of those 50. Then if you take 50, you can make this number of combinations of 9. That is huge number. But she did not do that. So she made 150 combinations of 9. And when you make these 150 combinations of 9, She tested them in the phenotyping center that we have. So then the plants are automatically scanned every day and monitored for growth. And then she did all these 150 synthetic communities of 9, and you have then effects on the growth. So there are synthetic communities that have a negative effect on growth, synthetic communities of 9 that have a very positive effect on growth. In this case, it's Arabidopsis, and everything in between. And here you see examples of how Arabidopsis grows with, let's say, more negative synthetic communities, and here with more positive synthetic communities. And she made a prediction model of that based on the composition and on the outgrowth, and she made a very well-functioning prediction model telling something about the composition of the 9 members of the synthetic community, whether they can tell something about how they would grow in the phenotyping center. And then she tested, let's say, 2 sets of highly positive, predicted highly positive synthetic communities, and a couple of predicted very negative synthetic communities. And then she tested, so these are combinations of 9 that were not tested in the 150, and then she tested them again. And then the vast majority of the predicted negative ones and the predicted positive ones were indeed also in the next round, indeed negative or positive. So from that we conclude that microbes are predictable. So from their genetic composition, you can predict whether they have a positive or a negative effect if you make these prediction models like this. So that was very comforting and very stimulating. So then we thought, okay, let's bring it a little bit to a higher level. Let's do it in practice. So let's select 100 Dutch potato soils on which last year potatoes were grown, and 100 European soybean soils on which last year soybean was grown. We collected those soils, brought them all to Utrecht University, made a soil bank, 100 soils from soybean, 100 soils from potato, and I will give an example of the potato. So here we have all the Dutch potato soils that we collected, 100 in total. And what the PhD candidates did, we call them the potato boys because they do the experiments. So we took these 100 soils and we wanted to know what's the effect of the microbiome on the potato. And in order to exclude the effect of the soil chemistry, we took 5% of the donor soil, and then of course of all 100, and 95% we had a gamma sterilized recipient soil that was for all the same. And then we grew the potatoes in these 100 soils and then tested them in the NPEC facility. And then they were growing like this. And then we tested, let's say, in the end stage, what is the effect of the different microbiomes of these 100 soils on the growth of potato. And then you can nicely see that there are potato microbiomes that have, let's say, a positive effect on potato. And you have potato microbiomes that have a negative effect on potato growth, and everything in between. And then, of course, you want to know, is this reproducible? So what Stijn and Jochem did, they took 5 best ones and 5 worst ones and reanalyzed them again in a new experiment. And then the result is shown here. Here are the 5 that were in the previous experiment positive, and these were the ones that in the previous experiment were negative, and they again were positive or negative. So there's a nice reproducible effect of the microbiota in those 100 soils, whether they have a positive effect on the potato growth or whether they have a negative effect on the potato growth. And of course, the next step, and this is all still unpublished, we want to make again a prediction model on the microbiome composition in those 100 soils, whether they can say something about growth of potato, and you know, we can identify microbes that have a positive or negative effect. We can identify maybe beneficial microbes to develop bioinoculants or biostimulants, but we can also, for instance, take the extremes and test the diversity sets of potato genotypes to identify genes of potato that maximizes the profits that the potato apparently has from its microbiome. So this is all ongoing, and this is of course something that we would hope to do in the future more. So soil microbes matter. They truly have a strong effect on in this case potato growth. We also want to do that for soybean. That's a little bit in an earlier phase. It's also a little bit more complicated because soybean grows, of course, much taller than a potato. So therefore, we have a 3D imaging center where you can identify individual plants, not only individual plants, also individual leaves. And you can measure stuff on that those kind of images. So you can also identify, well, which microbiomes have a positive effect and which microbiomes have a negative effect. And that will also be done in the near future. Okay, then I still have a couple of minutes left, I think. So I would like to have a little bit a different gear to showcase one of the other interesting studies that we do on potato. And that is work done by Dario. And Dario is interested in what is the effect of microbiomes from the wild ancestors of potato. So he is from Ecuador himself, and he is interested in, let's say, what we call reconnecting the beneficial plant microbiome alliances for future resilient crops. And he basically published recently a nice review on this topic in Cell Host and Microbe. So if you want to visit it, it's really nice to read, a very historical background as well, really nice. But what he did, he used Ecuador as a natural experiment. So he took soils from Ecuador, where he looked at combinations between cultivated potato soils, shown here, and he looked at soils that are next to it, like here, that are basically still native, untouched soils. And he took a soil from here and soil from there, and then he has in total 14 combinations from the north to the south in the Andes, 14 combinations of native versus agricultural soil. He brought them all to Quito. There he grew one genotype of potato in those 14 soils, or 28 soils basically, inoculated the plants with Phytophthora infestans, which is a severe disease. Then he looked for resistance. And what he basically found was that when the plants were grown in the native soil, they were almost always more resistant against Phytophthora than when the plants were grown in, let's say, the counterpart agricultural soil. So apparently there are microbes in that native soil that are not present in the agricultural soil that have a positive effect, or the other way around. The agricultural soils may have negative microbes in their soil. And of course, he tested that by looking at the microbiomes. And when he looked at the microbiomes of those 28 soils, so the native and the agricultural ones and the 14 sites, then you can already nicely see two clusters, one agriculture cluster in orange and one native cluster in green. So these are the microbiome profiles from those different soils. And if you zoom into that a little bit more, and you look, for instance, at these blue bars over here and the red bars over here, these are significantly correlated with either enhanced resistance or enhanced susceptibility. And all the blue ones are correlated with enhanced protection against Phytophthora, and all the red ASVs are significantly correlated with enhanced susceptibility to Phytophthora. So apparently, the native soil is enriched with microbes that are associated with enhanced resistance against Phytophthora, while in agricultural soil it's the other way around. And of course, you want to test, so in total 57 significant ASVs were identified that are correlated with late blight protection from the native wild soil. Then he wanted to know, does this really contribute to the resistance? So he did a soil transplant experiment where he used the live native soil that by itself has, of course, enhanced resistance against Phytophthora. And if you compare to the agricultural soil, you see more severity of the disease, which is basically the same as what I showed earlier. But you know, if you then autoclave that live soil, then this resistance is gone. Yeah? Then they become much more susceptible, the plants, if you grow them in the heat-killed native soil. The same also true for the agricultural soil. But if you then reintroduce 10% of the native soil into the heat-killed soil, you restore the resistance. So apparently there must be microbes in that natural soil that apparently contribute to the resistance. And interestingly, if you add the natural soil 10% to the agricultural soil, you also get this enhanced resistance back. And much less so when you do the same experiment with the agricultural soil. So from this we conclude that microbes from the native soil growing in the wild ancestor environment of potato can induce resistance against Phytophthora infestans. This basically relates back to the extended immune system of the plant. It needs microbes to enhance its own immune system. And with that, I would like to almost end and just showcase our phenotyping center one more time. Shown here, we have a very nice facility specifically for plant microbiome interactions. I already mentioned this above ground phenotyping facility. We also have a below ground phenotyping facility, especially by using plate systems to look at the root systems. And I quickly show a little movie on that. So here we see it. It's one of a kind in the world. It's a robotized center where we can automatically image root systems of plants, and it starts all with pouring plates. You don't have to do that by hand anymore. It's done by the robot. You pour the plates, they solidify automatically. We get automatically a sowing system, so the seeds are automatically sown on the plates. You can put 1, 2, 3, 4, 5, anything you want. And the plates are going into a cassette, and then the cassette goes into a growth facility where the plants grow automatically. And then the robot takes out those plates again and puts them in the different imagers. It can be an RGB imager, a fluorescence imager, a hyperspectral imager, to look at the root systems and how the plants grow, often in response to different microbes. And it can automatically be analyzed, and we can get all kinds of information about what is the effect of microbes on root system architecture, root colonization, production of coumarins. You can also add microbes in the system if you like, as you can see here as well. So if anybody is interested in using it, it's open for everybody. So just contact us. And a nice, this is the last slide, a nice image that I can demonstrate. For instance, you can automatically image the growth of a plant root system. You get AI-driven root segmentation, so you know what is the primary root, what is the lateral root. And if you use M-Cherry labeled bacteria, you can monitor the colonization of the bacteria on the root system, on the primary root, on the lateral root, and you can even quantify it in the system. So it allows you to investigate combinations of plant genotypes with plant microbes to see what is the effect and how can we optimize this all. And with that, I would like to thank everybody who contributed to this work, and they are all in this audience on the screen. And I thank you for your attention, and we'll be happy to take questions if any.
Thank you very much. That was a fantastic view of what you can do with these kind of automated systems, as well as soil transfer systems. So we've got a couple of questions in the chat. One from Tim George. Do you think a positive microbiome selected for potato would also be beneficial to soybean and vice versa?
Yeah, that is a very good question. This is, of course, something that we did not test yet. I think that, you know, the soils that we now collected were soils on which potato was grown in the year before. And the same was true for the soybean. The year before, the soybean was grown. But for potato, we already know you should not never grow potato in a row twice in the same soil. So potato is very vulnerable for the buildup for negative microbes or nematodes, stuff like that. So the only thing that we can do is identify microbial community or profiles that, you know, have a positive effect on the growth of the potato. But whether that can be, let's say, generalized that a positive microbiome for the potato is also positive for soybean, I don't think it will work like that. But you know, that needs to be found out, and nobody tested it.
Okay, thank you. And we've got one from James Goodman. When the donor soil was added to the gamma irradiated recipient soil in order to mitigate the effect of soil chemistry, is it possible that differences in micronutrients only required by the plants in small amounts could have influenced plant growth alongside the effects of the microbiome? Or is it a 5 in 95 dilution sufficient to mitigate this?
Yeah, that's of course something that we cannot rule out. It is possible that the 5%, of course, is enough to have a positive or negative effect on the plant growth. But that's, we tested different amounts. We tested 1%, 5%. We even tested slurry to take out all the chemical components of the plant, only the microbes, and spin them down and then mix them through the soil. But if you then look at the, let's say, how is the microbial community assembled on the plant in the test system, then it does not look like the original soil anymore. So it is a little bit of a let's say, on the one hand, as little soil chemistry as possible, but enough microbes to resemble the original microbiome. And of course, you cannot rule out that a little bit of the micronutrients that have a positive effect do the job like this. But we will find out if we later do the same thing with the synthetic communities, for instance. And for that, you know, the example that I showed you with the Arabidopsis and the synthetic communities, it was clear that, you know, different combinations of 9 microbes already have a strong effect on the growth of the plant. So we truly believe that majority of what we see is microbiome effects.
Okay, thank you very much. I think we've run out of time on this one, I'm afraid. I know, Simona, you've got your hand up. I did see you put a question in the chat, so I didn't know if you could just perhaps answer some of the questions. There's a couple more questions in the chat, so if you could just add some answers to the chat, that would be great.
Oh, yeah, I see it. Yeah, I will do it. Thank you.
Great, thank you. So Connor Fitzpatrick, have we got you online?
That's me.
To share? Right. So Connor Fitzpatrick is going to talk about the plant root microbiome during drought, spandrel or saviour.
Okay, that's right.
So
can everyone see my screen and hear me okay?
Yes, we can now. Yeah.
Wonderful. Okay, so first off, congratulations, Simona. We first met back when I was doing my postdoc, and I was just astounded by the research she presented at the MPMI meeting that year. And she was just a wonderful person to interact with as well, very supportive and encouraging. So very well deserved, wonderful researcher, wonderful person. Congrats. And thank you very much for allowing me to be a part of this celebration. Okay, so I'm excited to talk to you about this work that I've been doing for about 10 years. And my thinking on the subject has really been shaped by the results that have come out over time. Okay, so the very provocative title I've given is Streptomyces enrichment in plant roots during drought, Savior or Spandrel. Now, what is a spandrel? So in an architectural sense, a spandrel is just simply a byproduct of some architectural design choices. Okay, so when we look at arches in 2 dimensions, when 2 arches are joined together, we get this intervening triangular space. Okay, if those arches are arranged in a 3-dimensional shape where they're adjoining each other at right angles and a dome is placed on top, we get these really beautiful 3-dimensional triangular spaces, which are actually called pendentives, if you want to get super correct. And Stephen Jay Gould and Richard Lewontin, back in the 70s, upon visiting St. Mark's Cathedral in Venice, were just very captivated by these spandrels. Okay, and that prompted them to write their famous paper in 1979. So spandrels, the tapering triangular spaces formed by the intersection of 2 rounded arches at right angles, are necessary architectural byproducts of mounting a dome on rounded arches. The design is so elaborate, harmonious, and purposeful that we are tempted to view it as the starting point of any analysis. Okay, so one might be tempted to think that the spandrel itself was the purpose of this entire collection of pieces of architecture. Now, they applied this idea of the spandrel to evolutionary biology. And at that time, adaptationist thinking was very, very prevalent. Okay, so every trait that evolutionary biologists observed among organisms, one could create a just so story, an adaptive story, providing justification for the existence of those traits. And Gould and Lewontin really argued against this because multicellular organisms are structurally complex and built of many integrated parts. And so any primary adaptation must generate a set of architecturally enjoyed side consequences or spandrels. The number and complexity of these spandrels should increase with the intricacy of the organism under consideration. And in some region within a spectrum of rising complexity, the number and importance of usable and significant spandrels will probably exceed the evolutionary import of the primary adaptation. Now, enter the plant microbiome. So if we merge this concept of the spandrel, which is some beautiful, harmonious, elaborate trait as a consequence of adaptations occurring in the underlying architecture, then the plant microbiome maybe is some sort of spandrel, okay? It's an incredibly complex, intricate system, the plant and its associated microbes. And so any change occurring in the plant is almost necessarily going to bring about change in the microbiome. So, as a case study, is the widely observed enrichment of Streptomyces in roots during drought a spandrel? And this is the work that I've been doing for the last 10 years. So, let's go back in time. Plants migrated to the terrestrial environment 450 million years ago, yet first microbial life occurred 3.5 billion years ago. And so microbes have been present throughout all the adaptations required for land plants to persist and expand in the harsh terrestrial environment. And so a very simple question is, how are microbes integrated into plant responses to drought? Now, I'm going to go through this quickly because this literature has been around a long time, and my point is just there's a remarkably conserved compositional shift in the root microbiome during drought. And at this point, there are lots of examples, but I'm going to focus on 3 that all came out at about the same time in 2017, 2018. So the Sundar lab demonstrated in different rice genotypes that under drought, the actinobacteria become hugely enriched. The Coleman-Dare lab showed that this occurs across 20 different Poaceae species. And then my work showed that this occurred across a collection of eudicots and monocots. And since then, work has come out showing that it occurs in gymnosperms during drought too. And so if we look at all those instances, this covers essentially land plant evolution. We have gymnosperms and we have angiosperms. We haven't looked in bryophytes or ferns yet, but they don't, let's leave that aside. It occurs across a very wide breadth of land plant diversity. Now, drought enrichment in roots occurs generally across actinobacteria, but it's largely driven by Streptomyces. So here we're looking at the number of enriched ASVs across the rhizosphere or endosphere at different taxonomic levels. And so in the endosphere, we see the majority of enriched ASVs coming from the actinobacteria, and then the actinobacteria class, the actinomycetales order, and then finally the Streptomycetaceae family. And then in my work too, I found that although several families within the actinobacteria are enriched, because of their dominance in the root microbiome, Streptomyces is really driving this enrichment. And that's represented by these 2 bars here showing you the relative abundance of Streptomyces in roots across these 30 different plant species under watered and drought conditions, okay? Now, we've gone on to demonstrate that this enrichment is not passive, okay? So when we quantify the microbial load in plant roots experiencing drought, we don't see a huge drop in the total bacterial number during drought versus watered conditions. And so we interpret this enrichment of actinobacteria and Streptomyces as being active enrichment, okay? It's not just simply that they're persisting while everything is dying off. And finally, if we go back to the same soil, year after year after year, we find this very consistent enrichment of Streptomyces in roots during drought from that same soil. Okay, so this is 5 years running, going back to the same soil, we observe Streptomyces enrichment in roots during drought. And finally, across another axis of variation, we collected soils from across the United States, and we targeted locations that had really different chemical, climatic, and physical attributes. And we performed V3, V4, 16S sequencing of replicate Columbia 0 Arabidopsis individuals under standard watered and drought conditions in each of those soils. Okay, so here are all the soil locations. To the right of this map, I'm showing you an ordination of all the different chemical, climatic, and physical properties. And in this next panel, I'm showing you on the y-axis the log 2-fold change of different bacterial genera and the number of soils for which those genera were enriched or depleted during drought across all of these soils, okay? So we see mean enrichment of several different bacterial genera. So those are the ones above zero, and mean depletion of various genera across a number of soils. But the outlier here is Streptomyces. Okay, so this genus, on average, exhibits a log2 fold change of 2, and this occurs across 13 of our 18 soils. And I'm showing you now in a spiderweb plot the magnitude of that enrichment across all of our different soils. All right, so Streptomyces by far is an outlier in terms of the consistency of its enrichment across really different soils. And further, we can dig into the Streptomyces and look at individual ASVs, amplicon sequence variants. So these are just OTUs within the Streptomyces genus from our V3, V4 sequencing. And again, we find that a number, or by and large, Streptomyces ASVs are enriched in roots during drought. Most of them exhibit very restricted geographic ranges in terms of where they're enriched, with the exception of this ASV1, which is very widespread, and it's enriched across the majority of soils where it's found. All right, so this is ASV1. Okay, so let's go back to our case study and examine whether or not this looks like a spandrel so far. Well, I've told you that there's very high conservation across land plants. Okay, so every land plant we've tested to date does it. It's very consistent over both space and time. We have this large panel of diverse soils, and at some soils, we've repeated these experiments over time, and we find that it's highly consistent. I would go out on a limb and say it's the most reproducible host microbiome phenotype observed across plants or animals. I can't think of any other example where we see the exact same compositional shift across so many different axes of variation. So let's now revisit Gould and Lewontin's statement. So the design is so elaborate, harmonious, and purposeful that we are tempted to view it as the starting point of any analysis, as the cause in some sense of the surrounding architecture. And this was exactly my thinking a few years ago. Such conservation and consistency made me think that there must be some adaptive value to this trait, this enrichment of Streptomyces, because it is so consistent and we do observe it across so many different plant species. Now, let's get to the surrounding architecture of this, this trait. So, across this panel of diverse soils that I just showed you, we observe this strong relationship between the magnitude of Streptomyces enrichment in roots during drought and the pH of the soil. Okay, so at acidic pH, we observe low enrichment of Streptomyces in roots during drought, and then at high pH, we observe stronger enrichment of Streptomyces enrichment in roots during drought. And pH, as we all know, is very correlated with available iron. So under acidic conditions, iron is available, and under neutral and alkaline conditions, iron bioavailability goes way down. So that prompted us to investigate how the iron regulons, so the genes which are responsive to varying iron levels, are changing in response to drought. So here I'm showing you a heat map where each row represents a different gene in a literature-derived set of iron-responsive genes. Each column is showing you the mean Z-score of that gene in a particular soil type. Okay, so we have our 1 through 15 soils up at the top, and those columns are also color-coded according to whether or not plants are experiencing drought or watered conditions. And what you can see is that under the orange columns, we see a lot of purple, okay? Meaning that many of those genes in the iron regulon are being actively suppressed during drought conditions. And during watered conditions, they're generally being more strongly expressed, okay? So what we did is we took the differential between drought and watered condition, so the difference in expression between drought and watered, and we correlated that with the magnitude of Streptomyces enrichment. And we did so for genes across these different clusters shown on the right-hand side of this heat map. And that's what I'm showing you in this panel here. So each point here represents a single soil. Along the y-axis, I'm showing you the Streptomyces enrichment in roots in that soil. And then along the x-axis, I'm showing you that differential. Okay, so that, that score of suppression of that gene in response to drought. So negative values along the x-axis represent soils where those genes are being more strongly suppressed during drought versus watered conditions. And we see this fairly strong negative correlation where in soils where plants are more strongly suppressing genes related to iron uptake, we see stronger enrichment of Streptomyces in roots during drought. And I'm not even going to talk about it, but we found evidence that salicylic acid related immunity is also tied into this Streptomyces enrichment in roots during drought. Now, all of that is correlative evidence. So we wanted to experimentally demonstrate this. So what we did is we took soil from a single location, which exhibited partially acidic or mildly acidic soil, and we artificially added calcium carbonate and sodium bicarbonate to raise the pH. So when we do this, we can raise the pH to an intermediate level, so just hovering above neutral, and to alkaline soil conditions. And when we do this, we observe a sequential increase in the enrichment in Streptomyces during drought. Now, we wanted to know where this enrichment was coming from. Is it coming from under more alkaline conditions, Streptomyces are being enriched under drought conditions versus watered to a greater degree than in acidic soils? Or is it coming from under watered conditions, alkaline soils are leading to a depletion in Streptomyces? And that's exactly what we found. Under drought conditions, we see no difference in the abundance of Streptomyces across our different pH levels. So regardless of the pH, Streptomyces seem to be reaching the same abundance in roots during drought. And instead, under watered conditions, we're observing this sequential depletion of Streptomyces in roots as we increase the pH, suggesting that when we increase the pH, as plants are activating their iron uptake response, we're getting the depletion of Streptomyces under watered conditions. And during drought, when those iron uptake activities are suppressed, Streptomyces are able to proliferate. We did the same experiment, but now instead of altering pH, we added ferrozine, which is a very strong chelator of iron. And when we do this, we see the same thing. In the absence of ferrozine, we have this intermediate level of enrichment in roots, but not soil. And when we add ferrozine, we can increase this enrichment in roots, but not soil. And again, this enrichment is being caused by a depletion of the Streptomyces in roots under watered conditions when ferrozine is present. Again, indicating that when plants are strongly engaging their iron uptake response, this is suppressing Streptomyces. Okay, so that leads us to our model where under drought conditions, salicylic acid associated immunity and iron uptake are both suppressed, and that suppression is releasing the suppression of root inhabiting Streptomyces, which can then proliferate. Okay, so the suppression of iron uptake and plant immunity drive enrichment of Streptomyces in roots during drought. Now, we wanted to recapitulate these findings in a system where we could systematically add back in microbes under extremely controlled settings. So we needed to develop an agar-based system to test this. So what I'm showing you here are Arabidopsis individuals, Columbia 0, grown on different media types. MS is just our benign medium where the plants are very happy, and low water agar represents a media recipe that mimics what occurs in drought. And we know it does a pretty good job because the transcriptional response in roots in response to low water closely matches the transcriptional response that we observe across wild soils. Okay, so on low water agar conditions, when iron is replete, plants look smaller, they've stunted their growth, but otherwise they look okay. But when we drop the iron conditions to a limiting level, under MS conditions, plants are able to acquire that limiting iron because they can actively engage their iron uptake. But on low water agar, they cannot do that. Okay, they become completely chlorotic. And we can measure total chlorophyll content, which closely mirrors what we observe in the plants visually. And we can also measure shoot iron content. And here it's a little interesting because we see this drop in shoot iron content when we go from MS to low water agar, even at high iron, but this is cryptic because we do not observe it in the chlorophyll content. But when plants are grown on low water agar and low iron, they completely lack iron. And we can also show experimentally that on this media type, many of the components of iron uptake are shut down. So on this agar, we've embedded a dye, which is a pH indicator. So under acidic conditions, the dye is yellow, and under alkaline conditions, the dye is purple. And so one of the plant responses to iron starvation, at least for strategy 1 plants, is to pump out protons into their rhizosphere space in an attempt to acidify the rhizosphere, to make iron more bioavailable. We see that yellow halo of acidification in MS plants grown on low iron, but not on low water agar plants. We can also measure the activity of a key enzyme involved in iron uptake, which is called FRO2. And this enzyme reduces iron from Fe3+ to Fe2+, which is essential because the transporter of iron requires Fe2+, not 3+. And again, we see on MS media, under iron-limited conditions, an upregulation of the activity of this enzyme, but this is not present on low water agar. And finally, we have a reporter construct where we can observe the promoter activity of IRT1, that dedicated iron transporter in plants. And again, on MS media, we see this huge induction of promoter activity of IRT1 when we go from high to low iron, but that is completely gone in the low water agar system. Okay, and just to convince folks that this is actually real, we can measure shoot iron content in Arabidopsis growing in soil, and indeed we see a drop in iron regardless of the pH of that soil. And we also observe on our low water agar system a reduction in chlorophyll content with tomato and with rice as we go from high to low iron in MS and low water agar. Okay, so again, if we return to Gould's statement, we can apply this to Streptomyces. And so perhaps enrichment of Streptomyces in roots during drought is a consequence of the suppression of immunity and iron uptake, okay? It's not the, it itself is not the purpose of that sort of associated architecture, it's just simply a byproduct. Now, what about the functional consequences? Okay, so on this same media type, we can inoculate plants with different bacteria. And what we find is that some Streptomyces do have a remarkable ability to provide benefit to plants on this media type. So here I'm showing you an exemplary Streptomyces called Chaska 16, where on the low water agar at high iron, we observe an increase in biomass. Okay, so they're able to rescue biomass. And on the low water agar, low iron system, they're also able to completely restore chlorophyll uptake. And on this same system, we observe an increase in the enrichment of Streptomyces when we go from high to low iron. And we also see a reduction in Streptomyces as we apply our salicylic acid. So again, this just recapitulates the role of iron uptake and immunity in modulating Streptomyces abundance in plant roots. Now, at every single site, we have both enriched and non-enriched ASVs during drought. And we cultured as much as we could, and we recovered some of these enriched and non-enriched ASVs with different isolates. Okay, so we have isolates which match these ASV sequences. And here are profiles of enriched and non-enriched ASVs, where on the left, I'm showing you enriched ASVs from one of our sites called El Paso. And you can see that relative to soil, we see enrichment of these ASVs in roots under watered conditions and further enrichment as we go from water to drought conditions. Whereas non-enriched ASVs exhibit this flat profile. Okay, they're neither enriched in roots nor are they enriched in roots during drought. Okay, so we generated syncoms of enriched and non-enriched ASVs of isolates which match those ASV sequences across many of our localities. And we just simply tested whether or not any of them provide a benefit in terms of their ability to rescue biomass or chlorophyll on low water agar at either high or low iron conditions. And this is a very busy figure, but what I'm trying to show you is in white on each panel is the effect of the full syncom. In blue are the effects of the enriched ASVs in monoassociation, and then in pink is the effect of the non-enriched ASVs in monoassociation. Green represents the ability to rescue under low water agar and high iron, and yellow represents the ability to rescue low water agar, low iron. Okay, and I guess what's maybe important to focus on are the lines. So the dotted lines represent the non-enriched ASVs and the solid lines represent the enriched ASVs. And so if enriched ASVs are consistently providing a benefit, then those solid lines should always be higher than the dotted, and they aren't. Systematically, they are not. Okay, so microbial rescue is unrelated to a strain's enrichment status or origin. I think I'm going to just skip ahead to my final model because I've been talking too long. So returning to this case study, what we think is happening is during drought, plants are suppressing their immunity and they're suppressing their iron uptake. Whether or not you think that these responses are themselves adaptive, I don't know. I can't answer that. They just simply are what's occurring during drought. I think as a byproduct of these drought responses, we're seeing this enrichment of root inhabiting Streptomyces. Now, I didn't get to show you today, but what we have evidence for is that there's actually a lot of intragenous antagonism. Okay, so lots of interactions occurring among Streptomyces to vie for that open niche in plant roots during drought. And what we've been able to find is that the outcome of that antagonism is entirely decoupled from the ability to provision plant growth. Okay, so although during drought plants open up this niche that Streptomyces generally seem to be able to exploit, they also compete very strongly with one another to occupy that niche. And the strength of competition, it seems, entirely unrelated to the ability to provision any sort of plant growth promotion. And so returning to the idea of whether or not this is a spandrel, I think the field is very preoccupied with model 1 right here, okay, where we think that there's some plant gene which facilitates the reshuffling or reorganization of the plant microbiome, which then brings about some consequence. Okay, so we think that selection is acting on that plant gene via the effect of the microbiome. But I just want to plant models 2 and 3 a little bit into the field because I don't think they get enough attention, where actually plant genes in some trait which then controls the microbiome. And that trait may itself have adaptive benefit regardless of what's occurring in the microbiome. And model 3 could also be occurring where a plant gene exhibits this pleiotropic effect on both some plant trait and the microbiome. And the adaptation surrounding that trait is perhaps exceeding any benefit or antagonism occurring through the microbiome. All right. So anyways, with that, thank you very much.
Thank you, Connor. So we've overrun a bit. Thank you very much for that. But since it's Simona's day and she put a question in the chat, I will let her have the last question of the day.
Oh, wonderful, wonderful talk, Connor. Very, very good and exciting story coming up, hopefully. So why Streptomyces? I think I wrote the question before you started to go into the competition between isolates that are, part of the same genera. But why do you think Streptomyces? I mean, there are other bacteria, other taxa that are also sensitive to iron or sensitive to pH. What did I miss the point? Why do you think that Streptomyces are occupying this niche?
No, you didn't miss any point. I don't know, to be honest. We're still investigating this. Streptomyces recently have been discovered to exhibit this really interesting physiology called exploration behavior, where under certain conditions, they actually rapidly alkalinize their media in an attempt to kill off competitors. We've been trying to provide evidence that this is occurring in roots during drought, but we've kind of failed to find strong evidence to support that. What we do find is that across the isolates in our collection, Streptomyces seem to be really vulnerable to coumarins. So they seem to be very sensitive to the production of coumarins, which of course occur under iron-limited conditions. But yeah, we're still trying to answer that question of why Streptomyces. What's also kind of cool is Streptomyces as a clade seems to have really undergone diversification at the same time that plants invaded land. So about 350, 400 million years ago, that's when Streptomyces are essentially were born as a clade. So there is some matching there over evolutionary timescales as well.
Okay, thanks. Thank you very much. So thanks very much for everybody who put their talks into this afternoon session. I'm hoping Ken is still around, and I'll just pass back to Ken. We can't hear you, Ken.
That's the trouble when you have two mute buttons, one on your headphones and one on the software. So sorry about that. So yeah, no, we've reached the end of the day. Once again, the afternoon has been equally as fascinating as the morning. So yeah, all it leaves me to say is thank you very much to everyone for coming along. Thank you very much to the speakers. Thank you very much for the engagement. And also, yeah, it's been great to have this day, and also to honour Simona for an award. And all I will say is that I will put together the recording from the event and I will put it on YouTube. I haven't really figured out the best way to circulate a link to that, but we do have, obviously, we do have mailing channels, which is what's made all of you aware of this event. So we'll put it out through those same channels, and I'll also put it in on a LinkedIn approach, post. So keep your eyes open to that. We will, of course, be having the event again next year, and over the next 8 or 10 months, we'll be deciding who the next awardee will be. But for now, thank you very much, everyone. Thank you very much, Simona, for your very interesting and stimulating lecture at the very beginning. And thanks again for everyone attending and speaking. And hopefully we'll see you all again next year.